Hello, hello. It's working. Good morning, everyone. So let's start. We are sorry that we are delayed a little bit, but this conference obviously started at the same time as these manifestations and demonstrations around the university, so we were waiting a little bit so that people could enter. And it's my pleasure actually to welcome everybody here, but we will open officially the conference in the afternoon. So now we will start with the first presentation by my student Iago, and he will talk about the evolution of ludic phenomena. So now it's your turn.
Thank you, Arka. Thank you very much for everyone. So, as Yarka said, I'm going to talk a little bit about the evolution of ludic phenomena, focusing more on play and game, how they differ or are alike. And this presentation is based on the paper that we published this year in Frontiers in Psychology, where we try to show how sexual selection may have worked to make play a very diverse and polymorphous phenomenon. And if we look in the literature, we can see that there are basically two models of how play works. One model is more based on language like English, that treats play as a big trait that we can find in various behaviors. And game is just one subtype of play. Since game is played, game is also play, and therefore its functions, its evolution should be similar or at least equal to play. While the second model, which seems to become more popular with time, and I don't know why this is happening, but okay. Where we have a big trait that in Latin it would be ludus, in English it's usually used play. I use it playfulness because it's a word already used in the literature and to make it distinct from the play behavior. And then we have the behavior play and the behavior game, which they are alike in a certain part. There's a certain overlap between them, but they are different from each other. And most authors seem to suggest that they are not actually two different things, two circles. They are more like a continuous, where one pole is play, the other pole is game, and it's usually difficult to differ one from the other because everything is both play and game at the same time. It just the difference is in which side of the pole it is. So here's a table that we show in the paper, where we collected a lot of definitions of play used in the literature by several authors, both from evolutionary psychology, biology, social psychology, social anthropology, and so on. And as you can see, there are a lot of traits that can be used to describe play, to define it, and it doesn't seem to be a clear pattern of what it is play. And part of this is exactly because of this distinction, that some people use play as a trait and other people use play as a behavior. So we can, if you look in these two last columns, for example, we have two authors using six adjectives, 6 features to try to describe a play, and there's no overlap between them because even though they use the same words, they are not talking about the same thing, and this might be one of the reasons why the explanations of play, the literature of play, is so confusing many times. This other table is how people try to define a game, and as you can see, it's a lot more consistent. There's less features that people try to use it, and there's one that it seems unanimous. Everyone agrees that games have clear rules. So if you don't have rules, if you don't have clear rules, it cannot be defined as gaming, while play does not need to have rules necessarily. So here is a short list of phenomena that can be called play in certain languages and not in others. So we have free play, we have game, we have jokes, we have sport, music, and dramatizations, sexual intercourse, gambling, sadomasochism, puzzles, daydreaming. So as you can see, play can be used to describe a lot of things, very different from each other. So it's not surprising that we do not see a consensus in the literature about what is play, what is its functions, or how it works. So what exactly is play? Play is like Pinky from Pinky and Blaine. So I don't know if everybody knows it, but who is Pinky? Pinky is the one that every day asks, what are we gonna do today? Why? Because what we did yesterday is boring, is not fun anymore. So play seeks novelty. It does not like old stuff. You try to do something new every time. And as a consequence, it is spontaneous and aimless. You do not know where you will get. You can start doing one thing and you do not know where it ends. And it's always accompanied, it's always together with a positive emotion. And some people call it happiness, some people call it the flow, some people call it fun. I chose fun, I think it's better. While game is like brain. Game seeks challenge. He wants to dominate the road. It doesn't matter that we already did it yesterday. We'll try to do it today again, and if we fail, we'll try to do it tomorrow again. And when we succeed, then we'll go to the next level. We'll try to dominate the galaxy and then the universe. We are always trying to find new challenge. So it's goal-oriented, and it, as you saw, it always has rules. So you cannot try to do anything you want to. There's a protocol, there's a procedure that you must follow in order to achieve this goal, in order to overcome this challenge. And it does not need to have a positive emotion. It is the emotion that is together with game is training, is a kind of anxiety that you don't know if you will be able to succeed, if you will fail, or if you get what you want. So, and yeah, games can be fun, but they do not need to be fun. It's only fun when you overcome this challenge. And playfulness is usually treated as a trait, something that envelops all of this. And I tried to explain it the shortest way possible as the easiness to become intrinsically motivated. So one of the things that play and game have common is that they are supposed to be intrinsically motivated, as all of the other playful phenomena, like arts, like oracles, and so on. In humans and other animals too, but especially in humans, it is treated as an individual different trait. So some people are playful, other people are not. And it can also be used to describe activities and a property of things we do. In this case, it's usually called just play, but I call it playfulness to differentiate from the play behavior. And there's not a clear definition, but it's something like trying to activate the state of flow. Things that can easily activate the state of flow are usually called playful. And therefore, it can be present in almost any behavior. As long as you can get this intrinsic motivation, it can be described as part of the playful phenomenon. And this is not a new idea, something that I just thought this year and we tried to publish. It's very old. One of the oldest attempts to make this distinction is by the French philosopher Roger Capuchins, which described that play, or jeu in French, and can be divided in 4 categories, 4 primary types. The type of agon is and trying to get this motivation, this feeling of fun, of training through competitions, and therefore it's best represented in our game of skills. The second type is alia, which gets its fun or training from surrender to destiny. Try to not act, just see what will happen. So it's usually seen in religious phenomena, especially fortune telling and games of chance. Mimicry is trying to get this happiness or fun and training, trying to pretend to be something you are not. It's usually seen in arts, especially in dramatization, and in the pretense play of children and non-human animals. And the last one is innings, which is getting fun or training through the disruption of perception. So through things that cause dizziness, through drug use, watching terror movies, anything that can make you have an altered state of mind, of consciousness. And all of these types, I call them primary because they are not mutually exclusive. They can combine to form new types. And they are always also present in two modes, which are actually a continuum, an interval. So one pole, one side of this mode is paideia, which is this spontaneous or play-like way. And the other form is ludus, which is the structured or game-like form. So what we try to argue in this paper and in this presentation is that it's important to distinguish the play behavior from the play trait, which we call it playfulness, and which are not the same thing as game or as art or as religiosity, but they all have something in common. They all have something that make them be called play by a single word in some language. And playfulness, this play trait, might be this thing. But of course, none of these phenomena are unidimensional. So they are always playfulness mixed with something else. So we need to do more research and more cross-cultural research to make sure that this linguistic difference, they reflect what we really want to study. We are not mixing trait and behavior. We need to understand how these phenomena, they are related. So what we find in research about game is similar to research about music. Is it because both cause the same things, or is it because they have this playful trait in common? Also, interspecies research, because sometimes it's hard to understand how it evolved or what is function if we do not seek it in other species. Differentiate individuals that are highly engaged from low engagement, since there's high inter-individual variability. We need to understand better why it's so high in some people and so low in others. And of course, since they are not unidimensional, we need to always consider the various levels that they operate. For example, using Timberger's questions model as a framework. Here are the reference, and thank you.
If there are any questions, you can ask.
Thank you very much. As you know, one of the main evolutionary explanations for play is that it functions for learning across development. But if we think about learning, there's a typology of learning as well. So at least in humans, deliberate instruction, observation, vertical transmission, oblique transmission. So I'm wondering if we think about the typology of play crossed with a typology of learning, if we might get some insights that way.
one is mute.
Thank you for your question. Yeah, I didn't look exactly for these types of learning, but it's very possible that one of the reasons that play and difference between so many different phenomena might be because they use different types of learning. So, and it's a high possibility that as far as I know, no one has ever tried, but I think it might be a fruitful line of research if different playful phenomena can use different types, different ways of learning. I don't know. Anyone else?
Any other question, commentary? So thank you very much. Tocar o jazz. Eu já mandei Cristian perguntar. So let's continue with the next presentation by our student Timon. And he will be talking about cooperation and behaviors, talking about his project about with the method of twins, right? Twin studies.
Thank you. Hello, everybody. So I am Timon Le Baron-Kerif. I will talk today about cooperation and behaviors and present the study of my PhD thesis. I will also briefly present some preliminary results that we got from a pilot study that I realized with collaborators with the University of Sorbonne Paris Nord. And the actual thesis is with the University of Sao Paulo and the University of Paris-Nanterre. So the plan of the talk is basically here. I will start with defining the concept of cooperation and then talk about cooperative behaviors and finally use this introduction to present the study. So first, let's define cooperation because as we saw with the last talk, we need to define concepts. We can all have an idea of what can be cooperation or play or other concept, but once we need to define it, sometimes we found some difference between our expectations. For example, here, cooperation can be an umbrella concept. I can have helping behavior, I can have altruism, I can have collaboration or even moralistic interaction. And in the literature, from what I saw, there is no consensus and there is not much debate about defining cooperation and try to specialize this concept in sub-concepts. And so it has a drawback that sometimes the literature is difficult to relate. There is some result, but maybe they are testing helping and others are testing collaboration. But the good point is that when you study cooperation, you can choose your own definition and you can choose in the literature what suits you the best. So for mine, I wanted to go back to the roots of the word about cooperation. So co means together and operation means working, interacting. So here I took a definition from Garnier and Latour, which is working together with a common goal, yet not necessarily identical. I will explain this briefly. For example, with these two climbers, they are working together to reach the mountain top, which is their common goal. They are helping each other, but maybe one is doing it for the sport and the other one is doing it for the beauty of the scenery. So they are in the same goal, but slightly different, right? Once we have defined this, we are all on the same line here for cooperation. There is also different way to approach the study of cooperative behavior. The first and most basic one will be to do personal report, to ask people, are you cooperative and what do you do in this situation? But the big problem, of course, is that people are not always honest. So we shouldn't use this one much. On the other side, there is another design that was used a lot in the literature of cooperation, which is to use economical games. And this works much more. It is more realistic in the way that people, through this game, can earn money, can win money. And so there is a real incentive, and people want to win money, and so they will try to cooperate or will be selfish, and it will show much more honest behaviors. But there is, in these economical games, there is a variety of games trying to translate some situation of the life into games to see specific behaviors in specific situation. For example, there is two natures. There is the sequential resource allocation, which is you will invest or receive or give money in turns, and you will have many rounds. And so we can see at the investment or the total gain. And there is social dilemmas where you will make choices, but the consequences will depend on the choices of other people, and you will have to do strategies maybe to earn more. These games have different variation. For example, in sequential resource allocation, there is a dictator game, ultimatum game, trust game. And in social dilemmas, there is the prisoner's dilemma, which is very famous, public good game, and intergroups prisoner's dilemma. And inside each of these games, there are alternatives. So, for example, the dictator game has 4 alternatives, and all the others also. And we have a lot of games to use, and sometimes we can get lost in what game to use and how to interpret this. This is very, very interesting and very important. With these economical games, we were able to better understand the functions of cooperation. We were better understand to see the interaction and maybe to test the theoretical hypothesis of evolution of cooperation. It was very important. But it also has a strong drawback to me. It's that it, of course, is in Occidental and capitalistic view. And some studies try to do intercultural studies, and they try to adapt these economical games and not playing with money, but playing with some other resource that will be pertinent for the indigenous people, for the tribes. But the logic itself is fundamentally capitalist. It says that you will want to earn as much as possible. And maybe this logic doesn't apply to other culture. Maybe they have other view of properties, of goods, and of interpersonal relationship. So this is one drawback about other cultures, but also it can be differently interpreted by children. So it has been used with children, but if you replace money with stickers, for example, it can be an adaptation, but the interaction between children may be not structured in the same way than with adults. So it is, of course, interesting, but it has some complication of interpretation sometimes. So, well, if you don't use personal reports or economical games, what can you do? Oh yeah, something else also. Economical games, they, about the nature and the theory, they can also promote cooperation, but we don't really know what kind of cooperation is being promoted, because people want to gain as much money as possible, even if the group doesn't earn money. And so are they really working for the group? Are they really working together if the ending point, if the aim is very personal game, very individualistic behavior? And maybe this beneficiary effect for others, which is important in prosocial behaviors here in cooperation. Maybe these benefits for others are secondary effects. They are not wanted by the participants. So, well, if we don't use both these strategies, we can use experimental tasks. And basically, you take two children and you make them solve a puzzle, which is a much more realistic task. And it has a lot of good points for studying cooperation, for studying interactions. But the drawback here in the literature is that people always use a distant measure of cooperation. They always measure the consequences of behavior. For example, in economical games, they measure the total gain or the investment, which is the consequence of people's choice and action. And for experimental tasks, they will look at the success of the task. They will see the time the children will use to solve the puzzle, for example. And this tells us about cooperation in a certain way, but it doesn't tell us about the behaviors itself. And they have a somehow distant way to see cooperation. Because if we think about our definition, maybe two children are very working together with a very common goal, but maybe they are very bad at solving a puzzle. And so they will be very cooperative, but if we only rate the success of the task, they will rate very low on cooperation, which may not be representative. So these have been mostly used for studying the functions, but now if we want to study the mechanisms of cooperation and cooperative behaviors, we have one last option, would be to use experimental tasks, but to focus on behaviors and cooperative behaviors, which will be returned to the basics. And so here we are interested in understanding better what behaviors could foster cooperation, what behaviors people use in interaction, and what we could maybe learn from these behaviors. It will be very interesting also to see the behaviors and to see the difference that have been found already between groups, and to see what behaviors each group use. For example, there were difference between males and females, and between different kind of twins, and between the ages. And it will be interesting to see if each group that have difference in the actual literature also have a different behavioral strategy. That's the aim of the PhD thesis. So we aim at looking at behavioral differences in cooperation in twin children between 5 and 10 years old, to have an age range sufficient to make age studies, but also to have a big sample size, hopefully. And we will study this in Brazil, in France, and in Lebanon. And you can ask later for this happy choice of country. About the design, we will ask an online survey to the parents of the children. It will contain a zygosity questionnaire, which is very easy and can assess if twins are identical or fraternal, which is important. We also have a twin relationship questionnaire to assess the relationship of the twins, because they will have later a task together. Important to control for that and to explore it. And other eco-socio-environmental questions about the sex of the twins and the age and the condition of the family and else. But the important task for us is an experimental task that will be done at home in total autonomy by the parents. They will ask their twins to sit at a table and to do a drawing together. The twins will have to draw together their house with one single sheet of paper and 5 markers. And the parents will record with their cell phone during 5 minutes. And then they can send us the video and we can analyze it. The analysis will be principally on the time spent drawing. We'll look at the duration and frequencies. So drawing, co-drawing when both children are drawing together. Paying attention to sibling when one child is looking at sibling or at sibling's work. Physical contact, the time spent touching. And non-negative oral interaction when they are basically communicating without being aggressive or criticizing. We will also look at physical dominant contact, because conflict can also help to regulate the cooperation. So we will look at monopolizing when they deter the other twin from accessing resources, or aggressive acts, or negative oral interactions. The analysis will be done with Boris, and for presenting this result, we use a total of 13 videos that have been collected during a pilot study in France and in Brazil, but there were few videos, so we didn't distinguish the country in the analysis. It makes a total of 26 twins, but we wish for much more. And what was interesting is that the behaviors that we saw the most in these few videos was communication, oral communication in a non-negative way. They used it a lot, and closely from this behavior, they were the physical contact duration. And then insert, they were the drawing behavior. And all the behaviors were less frequent. What will be also very interesting is to have a bigger sample size and start to analyze behaviors depending on the groups categories, depending on the cultures, on the sex, and on the twin zygosity. And that's pretty all for the presentation of the study and the cooperative behaviors. Thank you for your attention.
Thank you very much. Very interesting talk. Do you have any questions, suggestions? There is one question over here.
Hi. First of all, thank you for your presentation. I was thinking about, I study personality psychology, and cooperative behavior is related to agreeableness. And we might think that people like to cooperate, but sometimes people who are too agreeable or too cooperative, they can build internal resentment if they feel they are being exploited by their peers or people who are not so cooperative. Do you have any thoughts on that?
Yes, that's what we were surprised sometimes to see in the videos, the conflict and the fighting even in very few of the twins, but they, as you saw on it was a very, I think 1 or 2 videos where the twins were actually fighting, still smiling because the parents were there filming, but actually, yeah, trying to resolve a conflict. And this cheating situation, if someone is too agreeable and too nice and too cooperative, and then will be used by others, is, I think it's a very huge theoretical aspect of cooperation, and that's why at first it had so much difficulties to understand the evolution of cooperation, because cheaters will be more successful. But so it was very interesting to study the literature and see all the mechanisms and the regulation, the threat of punishment, of retaliation, if too agreeable people retaliate and punish the cheaters, can be a mechanism. And it would be interesting to investigate it more because it has been mostly used in a theoretical aspect, and maybe it would be nice to see how it will work in behaviors also.
I'm wondering if you can comment a little bit more on the advantages and disadvantages of using a twin design here, because of course over human evolution, most people grew up with siblings, but those siblings were of different ages and different developmental stages, and of course here you have 2 siblings that are exactly the same age. So I'm curious what your rationale was and what your experiences were in terms of the advantages and disadvantages of using a twin design.
Yes, I would have really loved to talk more about the twin specificities. It's a very special design and unique design that we can use in the human population and also in other animals population. So there is 2 kinds of twins. There is the identical twins and the fraternal twins. The identical twins are almost the same individual, and at sometimes there is even an identity fusion. They feel more or less like the other. They feel like they are in their identity. And they share a majority of their genes, up to 90% and sometimes even more, of their genes. So it's, from a natural perspective, it's very strange to imagine that you have your own life, but if your twin survive and reproduce in future life, it is almost as good as if you live and reproduce. So the relationship can really, it can really impact the relationship. You can invest in your co-twin and you don't even need to be so selfish about yourself. And we want to study more about their relationship with a questionnaire that we address to the parents, because we want to see the particularity of twin relationship also and how it can influence. Because as you said, they are growing together and the parents have to share resources. There is always two children to care with the same needs almost. And so this is very interesting to see the influence between genetics and environments. That's why we use twin design most of the times. Because when there is behaviors in monozygotic twins, so the identical twins, and that there isn't in the dizygotical twins, which is fraternal twins, then you can suspect that there is a much more, a stronger impact of genetics in this behavior, in this happening, even if genetics and environments are always interrelated. But it can help in this way. Yeah. And then there is other particularities we would like to observe also the relationship between the parents and the child and the impact it could have because of this particular design of growing together and having to invest in two children in the same time. There is many specificities that the laboratory here at University of Sao Paulo is trying to investigate. Yeah.
Any other question? So why Brazil, France, and Lebanon?
Thanks. So Brazil, let's say, Brazil was Brazil and France were the basis because of the. I come from France and I've been here studying in Brazil, and so it has been an opportunistic possibility to study the two countries. But it was also very interesting because France has its own historical story, historical roots, and Brazil seems to have different cultural roots and history, and Brazil is also a mix of cultures that I consider that it's much more a mix of cultures than France. So it will be interesting to see if there were differences between behaviors in these two aspects. And then Lebanon was a third different culture with different roots, different origins, more Arabic origins. But in the same time, in Lebanon, I think there were some colonialistic influence from France, and most of people there, or some people there, speak French, and the questionnaire will be used in French there. And so we will see if the three countries have differences, and if they have not, if they have similarities between Lebanon and France, for example, because they have interactions. But also here in Brazil, there is a lot of Lebanese people because of the immigration, and I even heard maybe that there is more Lebanese people in Brazil than in Lebanon, but it's not scientific, I don't know about this. And so also if there is not differences between Lebanon and Brazil, it would be interesting, and that's a mix of possibilities between the cultures that I'm very curious to investigate.
Thank you very much.
Thank you very much.
So, do we have one more presentation in this session, or am I wrong? Okay, so we don't. So thank you for these two presentations. Now we will have a short coffee break, and then we will continue with the symposium on development. Okay, so enjoy the coffee break, and we will see you in 20 minutes in here, or 15 minutes. Izar and Nicolas Chaline will be presenting their research about comparative study of development and environmental effects. So thank you very much for coming, and it's your turn.
Thank you. Bom dia a todos e todas. Good morning. And so Yarka has already introduced it, so I have to get used with this. Let's see. Yeah. Well, so just to you understand what we are talking about here. So we are focusing on the developmental question and considering the environmental contributions and structure that channels the emergence of patterns of behavior. So we are proposing a dialogue considering some species, basically capuchin monkeys and ants. So I'm going to talk a little bit about the ecological approach, and I will present something about capuchin monkeys and learning the cracking behavior. Patricia will also talk about capuchins from Piauí state in the northeast of São Paulo, and Nicolau will talk about ants. Okay. So when you think about behavior and learning, we think about the context of environment, and thinking about humans, for example, we always think about tool use and objects and environment and things that channel behavior and actions and learning. Here I'm using the perception action perspective. You can see Professor Lockman in the picture. And he has a paper where he says that tool use is not something that pop-ups in the maturation of an organism of a human being. He was talking about humans. But it is a continuous phenomenon along development, and it's rooted on the perceptual learning. So subjects explores, manipulates objects, and then tool use is the result of learning the affordances of the objects and the tests. But we are focusing on capuchin monkeys learning how to use tools. Specifically, you can see a picture here of a capuchin monkey from Tietê Ecological Park. The capuchin monkey is cracking a Ciagrus nut using a stone which we call hammer. And the stone where the monkey places the nut we call anvil. And we have the social context also, and we are talking a little bit about it and how it influences learning. Something more about perception action perspective. I'm quoting here Esther Thielen, an important researcher. She was psychology, and she studied motor development in human infants. And she says that movement is not determined by the nervous system alone, but it emerges out of the confluence of circumstances. So movement is not in the infant in terms that it's not an instinct or something that is a fixed motor program or reflexes, but it's contingent on the environment. So we are using this idea to learn something about capuchin monkeys' development of motor behavior. Not only motor behavior, but how something more about nut cracking and the emergence of nut cracking behavior. This monkey here is from Fazenda Boavista in the northeast of Brazil. You can see that we was the monkey is also going to crack a nut there. And but the nut is different from the other that I showed you before, and the hammer is always different also. So these different objects, they impact in the way the learning acquisition will happen. And we will also use the idea of niche construction, and we have Leyland, Odling, Smith that work with it, but I like Richard Lewontin sentence that was, well, he said, living organisms are not passive entities. They actively build the environment and so the selective regime in which they live and reproduce. Meaning that when a monkey, we are thinking about monkey, let me return to the monkey. When the monkey cracks the nut, it leaves rests of the cracked nut and leaves the stones and leave marks and anvil substrate that may be a stone or a log. In this example here, it is a log. And we also have the social contest of cracking nuts. So when somebody starts cracking, the others usually are around. Some start cracking also, and so they are curious about it, and so nut cracking emerges, may emerge or may not. So they crack nuts. Here you see the example from Tietê Ecological Park, and here how do you use it? Let's see. I don't know. Let it be. So at Piauí. Some pictures showing you some details about the hand of the monkey, the nut, the stone, the size of the stone, the movement the monkeys have to do to crack the nut, and the social contest. You see a little one watching the proficient monkey cracking, and you see the little one here on this other picture exploring the cracking site. And here you have more examples. This picture, you can see the movement. You can see the tail, that it is used to give support, and how the monkey has to lift and then go down, and the knees, they have to squat. I don't know if it's the correct verb. And so, learning to use to use is not a simple thing, it's a complex behavior. And in social life, they have opportunities for learning. So this, you can see the Brazilian map over there, and the Piauí state in red. And then here you can see Gilboa City in the south of Piauí state. That's where we study these Capuchin monks that I'm talking about. And in the picture you can see a nut cracking site. I think it's important to notice that you have there some stones that were left by previous crackers there, and you have some concavities and holes in the anvil stone which guide how to place the nut. And so we think social learning is something broad. It's not. It's wider than only watching somebody cracking, but we have all this niche that was constructed and impacts the emergence of nut cracking behavior. These are some examples of the nuts they crack there. And we know that the reuse of tools that were previously used for proficient monkeys and the scrounging of the rests of nuts that were left there, they attract the other monkeys to the sites. And they are, so we think they are important elements to understand the emergence of nut cracking. In a previous paper, we it's called the fourth dimension of tool use. We are thinking about the importance of leaving these artifacts, these objects, in the nut cracking sites, and we gave some examples of how it could aid the learning of tool use in Capuchin monkeys and also in chimpanzees. So here this is a picture that I've already shown, but you can see the hand over there, the rests of the cascas. I forgot in English. You can see what is dead there. So they were left there, so young monkeys are attracted to these cracking sites. And where nut cracking is a tradition, and now we are talking about animal traditions, we see that they use the sites, they use what was on the sites to explore, and I wanted to highlight how they change the environment, and it canalizes the behavior of the next monkeys who will explore what is on this site. So more beautiful pictures, because we love monkeys and we think they are cute, we cannot decide which picture to use. And so in another paper, we found that the variability in actions, considering actions that are important in nut cracking sequence of behavior, we see that variability declines as the monkeys get older, and also as the monkeys get proficient in nut cracking. So it means that when they learn how to crack nuts, they focus their behavior on the more effective sequence that will lead to opening the nut. But now we wanted to study how do the use of stones for cracking and scrounging behavior develop. This is Juju monkey. I have to give the credit. One student of mine, Flavio, he created Juju monkey, so this is our pet from the lab. So we studied how objects influence the development of tool using skills in a wild population of brown capuchin monkeys from Piauí. And this is how it looks like. And we wanted to investigate the reuse of artifacts and the scrounging of nuts left by the individuals on nut cracking sites. Because we already knew it was important, but we didn't have a systematic study about it. Or we had, but not, we wanted to, we had more questions to answer. So this is published in Evolutionary Human Sciences. It is in collaboration with Andrés Ballesteros. It was his PhD. And with Patricia and Elisabetta Wieselberg and Dorothy Fragazzi. It's part from Ethosebus project, maintained by Patricia. And so we went to the field in 4 different field seasons, and we stayed in a place where monkeys are used to crack nuts spontaneously. We used 7 cameras that were disposed, as you can see in this draw, And 33 Capuchin monkeys arrived, and we filmed these monkeys spontaneously using the stones to crack the nuts and to explore also. And we used Boris software for transcription. 3 minutes, it means I must end in 3 minutes, okay. Thank you very much. So this is how it looks like. And we registered the frequency of stuns and nut manipulation and scrounging, if they were used before by another monkey, if they were reused immediately or 30 seconds after the previous monkey left, and we registered if the monkey cracked the nut or not. And we, considering that the objects left on the side attracts the monkey's attention, considering the variability of actions decreases of proficiency, we had the following hypothesis. The monkey's perceptions of nut as food item and stones as tools change throughout development as a function of the acquisition of proficiency in cracking. It means that when a monkey initially arrived on a cracking site and did not know that he could crack a nut, that stone over there would only be something from the environment. But after learning how to crack nuts, that stone becomes an artifact, becomes a tool. And so I'm going faster through these, our variables, dependent variables, and our independent variables. So we registered the frequencies of reuse, scrounging, when the monkey was cracking, when the other monkey had already left, and we had the age of the monkey, an index of behavior variability, and the level of proficiency of the monkey, if he was naive or not, proficient or not. We used generalized linear models to analyze, and our results were the following. So considering proficiency, behavior variability, and age, we found that the older, the more proficient, the younger, the more behavior variability the monkey had. And it was a confirmation of a previous study with a different population. But behavior variability was a better predictor of proficiency than age, meaning that what was more important here was not the age itself, but how the monkey was focusing on the task of cracking the nut. And we also found about delayed scrounging that the behavior variability was the best regressor, so it was positively correlated. So when the monkey was not focusing on the sequence of behavior necessary for the task, he used to scrounge more when other monkeys were not around. And about the immediate reuse of hammer and anvil, we saw that as individuals grew older, they decreased the immediate reuse of hammer stones. It means that, well, we can talk more about it later, and if you want, you can make questions, and then I can explain you in more detail. And about delayed reuse of hammer and anvil, we saw that objects used as artifacts in the absence of others. So, because we saw that the monkeys were reusing the hammer when they were more focused on the sequence of nut cracking, the best sequence of behavior used for cracking. So in conclusion, behavior variability was the best predictor, considering our hypothesis. Monkeys were increasingly likely to reuse tools as they were increasingly focused on cracking behavior. And we think that objects become artifacts or tools as nut-cracking behavior develops. And the social context establishes the elements of the cognitive niche marked in the physical world. And the process of cognition results from the interactional dynamics involving nuts, stones, and each animal, what each animal can do, and considering its ages and experiences. This is just, I just wanted to show you, if you want to learn more about Capuchin monkeys from Fazenda Boavista, you can find it on YouTube. And that's it. Thank you very much. Thank you for people who helped in the field, for everybody who funded us, and well, that's it. And that's it. So, wow. I think we can move to Patricia, and then Nicolas, and then we sit over there, and then we have all the questions. Okay. Então, acho que é. Eu estava falando tranquilamente, eu nem vi que ela estava. Não sei se é disso, gente. Esse não tomou essa água, né? Não, não tomou. Posso tomar, porque eu tenho sede. Gente, eu não sei se é disso que eu acho aqui. Ah, isso.
Quer ver se você consegue colocar a apresentação eletrônica para você começar?
Põe, pode pôr. Esse negócio. Como que é esse negócio?
As setas mudam os slides e o botãozinho vermelho. É a primeira? Aqui, né? Foi?
Pode apagar a luz aqui também, né? Good morning, everyone. Bom dia para todo mundo. Thank you for being here for our symposium on development. And today I'll talk to you, I will briefly summarize my main research on social systems of Capuchin monkeys, and then I will talk a little bit about a set of studies on the development of these social systems or their social behavior. So I don't know if everyone is familiar with the robust Capuchin monkeys. Of course, Bri has just spoken about the Capuchins in Piauí, but in fact, Capuchin monkeys or the robust Capuchin monkeys refer to nowadays to 8 species of the genus Sapajos, and the entire genus occur in all biomes of South America, and they occupy areas with very different ecological features, and they present correspondent variability in their behavior. There has been a lot of studies investigating their plasticity, a lot in their, in their modes of locomotion, and especially in their feeding behavior. But in our group, my study group, we have been interested in the variation of their social systems in relation to the ecological features. And we have shown that features of social organization, mating systems, and social structure reflect differences mainly in the risk of predation and the features of food sources they use. In particular, group size and sex ratio vary in relation to predation risk. The dominance hierarchy, dominance relations among females vary in relation to the features of food sources, and the dominance relation between the sexes, between males and females, reflect differences in demography, in sex ratio especially. But today, I will focus on the comparison of two populations that me and my group, we have been studying for more than 15 years now, 12 years in Bahia and in Piauí. Briseida just spoke about the population of Fazenda Boavista that we study in Gilboés. This is the northeast region of Brazil. It's an area of the ecotone between Cerrado and Caatinga. So, as you have seen in the pictures that Bri showed, it's a kind of dry environment. And the population of Sapajos Santosternos that occurs in the south of the Bahia state. So it's not northeastern region, you said northeastern of São Paulo, but today I will talk about these two populations of the northeastern of Brazil. One that occupies an area of Atlantic forest, and one in the more xeric environments of Cerrado and Caatinga. So I will summarize our main findings across all these years. So we have shown that group size in Una is much larger than in Boavista, and this reflects the fact that there are more females in the groups, more females associating groups in Una than in Boavista, but we also have proportionally more males in Una than in Boavista. So we have higher group size and sex ratio in Una, the area of Atlantic forest, than in Boavista. And this happens in spite of similar conditions of food availability in both areas. In fact, here we have food availability in Boavista and overall similar to Una, especially due to patches of plantations that occur within the Atlantic forest, plantations of cocoa and jackfruit, and then the oil, and the monkeys feed a lot in these trees. So in spite of similar conditions of food availability, we have differences in group size and sex ratio. This, in the literature, usually is related, especially the higher sex ratio, to a higher perceived risk of predation. What is exactly what we see when we compare both populations based on the frequency of alarm calls that we record that the monkeys emit. So monkeys in Una, they perceive, based on their alarm calls, they perceive more or less twice the predation risk than in Boavista. And this is because of the presence of the arpia, which is a super large bird that predates the capuchins, but also due to still hunting by humans. They give alarm calls for signs of hunters, such as dog barks. We have also shown that, esqueci de marcar meu tempo, né? Eu ia marcar, tá indo tudo bem? We have also shown that the dominance relations among females in Boavista are nepotistic, are linear, nepotistic, very stable across years. And in Una, we almost cannot see dominance relations, just for a few. The dominance hierarchy is almost absent in this area. Again, in spite of similar features of food sources. However, we can see here that in Una, more members can feed at the same time in the same tree. probably when they are using those jackfruit and the oil trees that I told you about. Regarding grooming relations, in both areas, in spite of the dominance hierarchy in Boavista, we don't see any relation between dominance and grooming relations. Relations are reciprocal, equally reciprocal in both areas. The main difference is that in Boavista we see a slight modularity, while in Una everyone grooms everyone, so it's a super dense grooming network. And finally, we have shown recently that the index of female dominance over males in Capuchins varies according to sex ratio. The higher the sex ratio, the more dominant females are in relation to males. However, you can see here in purple that in Una, this female dominance is below the expected by this relation. So overall, when we summarize this data, we can say that in the population of Una is kind of more egalitarian, relaxed, and more affiliative than the population in Boavista. We then turned our attention to understand whether these differences are a consequence of an innate temperament, if these species are different because some kind of genetically inherited difference, or if we could identify differences in the developmental trajectories of these populations within these different contexts, social contexts where they are growing. For that, we started to, in 2013, almost 10 years already, we started to film every newborn in each of these populations. We tried to have one day, one focal filming day per week from the first week until they complete, they were 3 years old. So 36 months, filming 1 day per week, every week. Of course, we have a lot of data missing, but this is what we tried to do. And we have babies here in both populations. So I'll first show the work of Irene Derval that is sitting there. She was my PhD student, and I'm reporting here her incredible work. Investigating the personality traits of the newborns or the youngster, the initial development of 12 individuals from Una, the Sapajos Xanthosternos, the egalitarian population. She investigated 9 males and 3 females, and from all that video database, she selected 10 developmental points, the first 3 months, then the 6th, the 9th, the end of the first year, the end of the infancy, and then 3 points in the 2nd and 3rd years of life. the time goes very fast. So she had almost 5,000 minutes, and then from the 44 behaviors she selected the most frequent 15, and with that she conducted a principal component analysis, and she found 4 behavioral axes that we called sociability, that is mainly related, several features here, but it's very importantly related to the time spent with neighbors around, anxiety that is relating to eating and scratching, openness that is related to play behavior and sexual behavior. In young, she was studying individuals until the 3rd year of age, and activity that is related to moving, walking. We can see here that individuals varied a lot across the developmental points in these 4 traits. She found overall certain repeatability or significant repeatability in sociability and anxiety. It's too small, I don't know if you can see. But what is important, she found this significant repeatability, but values were below 0.4, so it's kind of not so repeatable. Repeatability is a measure of the rate between intra-individual variance and inter-individual variance. So when we say repeatable, it means that the individual is consistent and there are consistent inter-individual differences. And we can see here it's showing what is the months where this repeatability is significant. So we can see that in sociability, we have repeatability in the first year of life, and then it disappears to reappear in the third year. For anxiety, we also have this variability in the first 3 months. This is not appearing yet. Nothing almost for openness, and anxiety also varying across the developmental points. So they are very unstable in this beginning of their life. So we say that personality is not established yet in the first 3 months, 3 years of their lives. The same, more or less the same result. Oh, I forgot to, the first 3 years, I forgot to correct this one. So Julia Omena did her master's following more or less what Irene did with the population of Una. She studied the monkeys in Fazenda Boavista, but looking only to the sociability X of personality. And she was looking to the initiation of social interactions, social behaviors by the immatures to try to investigate whether this social personality would be established. She repeated the procedures that Irene developed for the study in Una. She did not also find repeatability. Only she found only repeatability, significant repeatability in the 2nd month of life and the 9th month of life. So again, this we cannot say that they have a personality, consistent social behavior yet in their first 3 years of life. Then these 2 other studies, 2 master studies, Marie Caroline, Franco Rogério, and Emily Faverin, they studied the development of the association network in these 2 populations. They used the same video database, and they recorded the neighbors within 1 meter of the individuals, and they developed the social networks and compared the development of these these network metrics, degree, strength, density, and modularity. And Marie also found this great variability across development. And so that we could only see a significant difference in strength between the first and 2nd month of the individuals and the other developmental points. She found no significant difference in degree, density, and modularity across the first 3 years of life. And this difference in strength disappears when remove the mother from the network. So we saw a stability in social development across the first 3 years in Boavista. Almost there. I know I'm late. The same thing Emily did with the population of Sapajos Chantosternus, the same measures. We see even more variation here. But here, Emily had to reduce the developmental points. She had more developmental points than the individual, so we conducted a cluster analysis, and we compared the development across these clusters of development. So we have 3 points for strength and 4 for degree and density. And again, we find a significant difference from the 3 first months of life and on. So we don't see a great difference in the development, the social style of both populations, but development seems to be faster in Una in comparison to Boavista if we consider this difference in the first 3 months of Una in comparison to the rest of the first 3 years, while in Boavista they are always more or less the same. And we attribute this difference in the rate of development, in the pace of development, Flavio, we don't attribute this to inherited differences, but to these, again, to the difference in predation risk. It's common in primates, even in human primates, that in conditions of higher perceived risk for the babies, mothers promote independency, earlier independency than in areas with lower risk. So sorry for going over my time, and that's it. Thank you very much. Thank you all the students and field collaborators, the funding agencies. Thank you for being here.
Right. Good morning to all. Thanks for coming to this event. Thanks for the organizer for inviting us to present some of our research, which is not about human animals. So when Resende proposed that we did this symposium, I was thinking, what could I talk about? And I thought that some work I did some years ago about how development is influenced by the rest of the group would be something interesting, because there is some concept associated to that which actually can be applied to any living organism, more or less. So I will present my work on the clonal ant, Oceraya biroi. The idea behind this research actually is, although development has become a fundamental study area to understand behavior, and it must be obvious to who works on humans, psychologists, it's less obvious perhaps when you think about invertebrates such as ants, insects, and even microbes. What happens is that people working with humans actually sometimes they get a bit set back by some ethical problem, by the facility to use this organism in controlled experiments, which in ants we can actually set up pretty much whatever experiment we want. So it's something which that we take advantage of to do research on some more complex mechanism associated with development and social behavior. And last, before I start presenting the work on ants, I think something we may not always remember is that our environment, and perhaps the most important factor in the environment that will influence us, is other organisms from the same species. So in humans, we interact every day with lots of people which are not related, which are work colleagues, and surely this is something that will influence our future behavior during infancy as well. This surely will influence what adult we will turn. And this is something that actually, in my opinion, is true of whatever organisms that interact in groups, is that the proper interaction creates an environment which actually also modifies the way the environment is perceived by the organism through the filter of the social group. So I think this is one of the important messages during my talk. So before I start, I will just present 3 of my collaborators in this project. My 2 PhD students, Serafino, Emmanuel, and Daniel Kronauer, which is now in the Rockefeller Institute, University in New York. And he's continuing work on the genomics of social behavior using this same clonal radar end. So if you're interested in this type of work, look at the website of Daniel. He's publishing loads of stuff on genomics in ants. So at the basis of my research actually is cooperation and conflicts. So you all know that Maynard Smith and Satsmari, when they work about the major transitions in evolution, they saw that the complexification of organisms from genes to cells, cells to multicellular organisms, and multicellular organisms to societies, which actually have division of labor, which have a difference between reproductive individuals and not. It's something that is important to the success of organism, but it's something actually we have to deal with as biologists, because also it seems to bring some advantages. There's also a lot of existing conflict that comes with the fact that stuff is living together, be it genes, cells, or organisms. So these conflicts, which are one of the mainly studied problems in social behavior and social evolution, is the fact that societies are heterogeneous, and the interests of individuals inside the societies can be different. Actually, between societies as well, you can have conflicts. And within individuals, it's well known that cells can conflict through presence of different genetic materials, say the organism, and also genes will compete to get to the next generation. So it's all levels of conflict. And surprisingly, actually, the concept that will decide who wins, who will pass on their genes, who will be on top, actually very similar according to the different levels of complexity. For example, I don't know if you are aware of this famous classic example of the social amoebas, Dictyostelium discoideum, sont aqui amoebas that actually live in a solitary form in the environment, and when resources are lacking, they actually group together from a slug. The slug will go away from the environment, and then it will sporulate, so that there's a resistant form of the amoebas will be dispersed to hopefully a better environment for the cycle to start again. So what people have been, John Strassman principally have been studying is how genetic heterogeneity inside the slugs will influence the phenotype of the sporulating body. And what they found is that when the slug is clonal, that relatedness is high, actually, the sporulating body has got a longer stalk and less spores. when there is more than one clone inside the slug, the stalk will be shorter, and more of the cells actually will differentiate in spores, which means that collaboration is lesser. It seems that there is a conflict between cells of different genomes that makes that the cells cooperate late, so they will make a smaller stalk, and also more of them will differentiate in spores. So more of them actually wants to get to the next generation at the cost of other cells in the slug. So we have the similar systems in our complex animal societies, but it's a bit more difficult to study what is going on there. And mostly we lack genetic control of the composition of the control of the genetic composition of the groups. Something important, but which must be obvious to you, is that genetics is not what decides what's going on at the level of the group, at the level of the behavior. So social hierarchies, which are actually also very widespread in social animals, such as primates, but ants as well, and also in other social species, such as this fish, where complex behavioral interaction actually decides cooperation and the result of cooperation. So our model is a clonal radar hand, Ocera biroi, which is unique in ants because it's obligatorily parthenogenetics. So actually, workers will lay unfertilized eggs, which give rise to more workers. There are no hierarchies, so everybody in the social group is reproducing, and they are thus clonal. Most colonies actually are composed of identical individuals who have the same genome. And they are just an ideal model for studies of development in animal societies. They also have an army ant cycle, which means that in the colonies, there is a strict difference between the reproductive phase where eggs are laid and the workers actually are in the form of a pupae, and the foraging phase where the eggs hatch and the workers will go out of the colony to forage. We pass this. So this is what a colony of Oocyarea looks like. You see here all marked workers, a larvae, so this is the foraging phase, and the workers will go out to hunt for other ants to feed the larvae that will develop into more workers. So what we start doing, looking at the control social group which have the same genotypes, the same age, is our experience as the experience of the environment which will change behavior. So we did actually a pretty simple experiment. We had a nest chamber here where the ants are, and several times during the week we actually opened the nest for them to go out to forage, and we assigned the individuals to two experimental groups, one which will actually encounter prey during the foraging, its foraging effort, and another which will not encounter prey, so which will be unsuccessful. We did that for a whole foraging cycle, which is 15 days, and what we observed actually is that the workers which were reinforced with prey actually took less time to go out as we make them do more sessions, and the unsuccessful one actually would stop going out or would last a lot more before going out. And when we looked at their behavior a month later, when they were foraging again, we saw that there actually spatial segregation between the workers that were successful or unsuccessful during the first set of experiments. the workers which were successful actually were closer to the exit of the colony, and they were less in presence of the larvae. So what happens actually is that workers that were successful were going more outside, and they also are in a place in the colony which does not have larvae. So actually they are more successful at foraging. They stay more away from the brood. So workers which were unsuccessful actually come smaller to the, closer to the brood, which probably is another reinforcement for them to stay inside the colony to take care of the brood. So you have division of labor based so on the perception and the experience they have of the environment and the social group, which is something that actually had not been studied before or shown experimentally before, which shows a strong effect of the environment on the behavior, on the future behavior of workers. So I don't have much time to talk about this. Let's see. So what happens in this species is that actually there are two morphotypes. A morph which is more highly reproductive, which is this one that are more similar to queens in other species, and one which is less highly reproductive. In terms of number of ovarials, so their fertility, these high individuals are actually potentially more fertile than the low ones. And what we did actually to study how this was regulated in the social group, so who reproduces in workers which are actually genetically identical. Let me remind you. We actually mixed different clones of the same colony to see how they would react to the presence of competition inside the social group. So in a previous experiment that we did, we showed that actually the fertility of the social group is regulated through the proportion of highly reproductive individuals, which means that when there is a lot of highly reproductive individuals in the group, in the next generation, the social group will produce less of these individuals. And on the contrary, if there's a small proportion of highly reproductive individuals, you will have in the next generation a higher proportion of the individuals. So there's a flexible adjustment of colony fertility through which is proportion dependent on the proportion of fertile individual in the group. So what we did, we used the principle of the chimera. We mixed genomes of the same species to see what would happen. And we predicted that actually because of conflicts, the clonal line would actually try to overcome the other clonal line and produce more reproductive individuals. So this is the experiment. We actually mixed these two clonal lines, clonal line B, clonal line A, and we have control lines to see what would happen when we mix them up, to see how the social environment and potential conflict linked with genetic heterogeneity would actually influence how the group does and how each clonal line inside the group would do. So what happens is that we have here pure colonies of clone A, pure colonies of clone B, and our chimeras, which are a mix of the two. What's interesting is that one of the clonal line A produce a lot more individuals than the other one, and the mix. So it seems that actually conflict inside the colonies made the colony less fertile and, in the colony. What happens in the chimeras is that clone line B actually outcompeted the other clonal line. So actually we showed a conflict which actually was resolved always in the same direction, with clonal line B winning on clonal line A. And what happens is that in our clonal line, in our chimeras, there is a lot more of clonal line B which develop into highly reproductive individuals. Oof. So what happens actually, and this is the message of this experiment, is that clonal lineage A has a low inhibition of development of its larvae in highly reproductive individuals, and these individuals in return also have low probability of developing in highly reproductive individuals. On the contrary, clonal lineage B has a high inhibition of the development of larvae in highly reproductive individuals, and the larvae themselves have a high probability of developing in a highly reproductive individual, which means that actually there's a calibration in clonal line of the inhibition of workers on larvae and the probability of larvae to become highly reproductive individuals. When we switch that, ¿qué está acontecendo? Okay, tá bom. What happens is that when we put larvae of the clonal lineage B in the adults of clonal lineage A, inhibition is low here, and the larvae have high probability, so the clonal line B will produce many highly reproductive individuals and actually behave as a social parasite, exploring, I can't remember the word, taking advantage of the other genetic line, whereas clonal lineage A actually cannot develop in highly reproductive individuals when they are raised by adults of the clonal lineage B. So what happens is that colonies are calibrated for the group functioning. Also, they seem to be calibrated to certain environment because this clonal lineage A is very fertile in constant environment, and this clonal lineage B is less fertile, but actually is more resistant to lack of resources because these highly reproductive individuals actually live for longer. So this is, to finish, I think it's a nice story that we showed that the genome of the individuals in the group you interact with actually will influence what you become during your development. And when we talk about ecology, we see also that in a species of clonal, which have clonal lineage, actually different behavior, different reproductive phenotype can develop, which actually can lead to parasites and perhaps the evolution of new species when you mix actually this clonal lineage together. Well, the conclusion, the environment influence the development of alternative behavior in social species and probably other social interactions, social context is of crucial importance. The interaction of genome present in different individuals influences the own individual gene expression and phenotype. So this complex gene-gene environment interaction can be a general factor, and more importantly is that the individual, as they experience the environment, will change their behavior, and in turn will change the way they feel about the environment. So their feedback loop constant about the experience of the individual, which comes from the environment, and the way it will change its behavior with regard to the environment it is confronted to. Okay, excuse me for going over time. A lot of people actually contributed with discussion, with work, and also it's a chimera because some of the photos here actually are not people from the lab. I'll let you guess which one they are. And I will thank you for your attention.
So, thank you very much for a very interesting symposium, and I think very relevant for evolutionary thinking and psychology. So, if there are some questions, you can ask. Oh, here, sorry.
Hi. I have a question for Patricia. You mentioned a relationship between food type and social hierarchy. Can you talk a little bit more?
Thank you, Mauro Jr. And the features of food sources refer to a quality that we say that turns that type of food source monopolizable or usurpable. So it reflects the size, more or less the size. So if it has to be large enough for more than one individual share the same food source for a period of time that will make possible for another one to try to claim and to displace that individual from the source. So it's a type of source that generates direct competition. So usually we say that it's a type of clumped food source. So if it's too small, so I can eat this so quickly, nobody will steal it from me. If it's too large, all individuals can feed at the same time, so there is no reason for direct competition. So it's the size and the amount of food that make it possible to control, but also someone try to displace you to have the control of the food source. Did I explain? Thank you.
Other questions?
It's for Resende. You mentioned in your talk that less behavior variety predicted more proficiency at nut cracking. Now, I thought it was interesting. How do you explain that?
You thought it was the opposite?
Interesting.
Interesting, right. So what the index of behavior variety was built considering the frequency of behavior they were doing at the nut cracking site, because we were filming what they were doing there. So they could take the nut, take the hammer, and also do something with the other guy, try to play. They could do many things there, but what they should do to be a proficient nutcracker was take the nut, position the nut on the hammer, on the anvil, take the hammer and pound it, and that's it. So take, place, take, pound. The more proficient the monkey, the more focused on only this sequence of behavior they were. And the less proficient monkey, they did many other kinds of things. And so this behavioral variability was considering only the nut cracking task. So, and so when they were, they learned how to crack nuts, and they got to the nut cracking site for cracking, let's say. So that's what they did.
Some other question?
I'm wondering if you can, on the clonal ant species, if you could tell us a little bit more about the chromosomal structure in that species? Is it diploid? Is it haploid? Is it haplodiploid? And then also, how unique is this species? Are there closely related species that are also clonal? What explains the evolution of this clonal reproduction system? What were the ecological factors that led to the evolution of this type of reproductive system?
Right, and well, these social group where they are all females, they actually are diploids, and the parthenogenesis occurs through central fusion during meiosis, which is something studies which have been done to actually determine how clonality works, which seems to reduce actually homozygosity in the descendants. So it's one of the mechanisms that actually makes that the load of clonality is not that high, and also the fact that they are social. There is two aspects of these ends which actually may be an element to explain this reproductive system. The first is that they are distant, well, they are relative of the army ant. And army ants, they all have a syndrome where they have cyclic reproduction, where the eggs are all laid at one point, and the colony will enter in a statary phase. So the workers actually do not go out during this phase. And when the eggs hatches and larvae are present, they will actually go out every day to look for prey, which are other ant species. I don't know if you've already come across army ants in the neotropics or in Africa. Something that is really impressive. So the Sarapakis has got this syndrome at a lower level because colonies are less numerous. The explanation about the fact that clonality was selected is that they actually occur originally in parts where there's a high monsoon. Is that right, monsoon? So actually, sexual reproduction, which needs a reproductive to go outside, have nuptial flights to meet, and then individual queens to found a new colony. This is actually very, the probability of this to succeed actually very little. So it probably, because of this, that some mutants which would reproduce parthenogenetically were selected in this species. And then you have the problem of what a species is, because so we have clonal lines. Actually, Daniel has discovered 4 different clonal lines, so actually they could have evolved separately, and so it's something that is really, there is some, these are the hypotheses, but we actually cannot really know. There is a lot of species, there is few cases of species of ants which actually also are clonal and which comes from the same geographic area originally. So it's something that could have been selected repeatedly because of the difficulty of sexual reproduction in these environments. Okay? Thank you.
For the second talk on the development of skills being higher in high predation monkey populations, I'm wondering if an alternative hypothesis is that if you're more likely to die young, you reproduce faster. And so I'm wondering if the skills that are developed are maybe more reproductive skills or more survival skills, if that's a good alternative hypothesis. Do you have thoughts on that?
It's a good hypothesis, but in fact, although they promote independency earlier, they don't reproduce faster. The interval between survival offsprings, so it would be interbirth interval, but is higher in Una than in Boavista. So it's not exactly what we would expect. Thank you for the question.
Hi, so my question is about tool use in Capuchins, and I was wondering if you recorded any data about reproductive behavior, maybe number of offspring produced or opportunities to mate, because it occurred to me when you were talking about how tool use is such a social event, I'm wondering whether, for example, males, those who, I guess, develop proficiency more quickly might be chosen more often for reproductive opportunities, because that might indicate intelligence or ability to adapt to new circumstances. And that, I guess, would maybe reveal something about the development of how tool use would evolve and perhaps be sexually selected in some ways. So I don't know if you have thoughts, if you have that data or you have thoughts on that.
We have data about the reproduction, and we have been following these monkeys for a long time. But we were not focusing on this, but we believe that the social, what we noticed is that the monkey is not paying attention in a particular monkey. It takes the opportunities for learning. So the young monkey or the naive monkey, he stays nearer the proficient monkey if he's tolerated. So and sometimes when they grow up, they are not tolerated anymore. So we have this thing, but we also have, for example, adults together in the nut cracking time, let's say, and we have some scroungers who have the possibility to, or the opportunity to scrounge because they are tolerant. For example, when you have an adult male cracking nut, and then it comes another adult male for scrounging, he will not be able to do that. Okay, so he will be beaten. He will be attacked. But an adult female, perhaps she will be tolerated. But if she's in estrus, I don't know if you are familiar with capuchin monkeys in estrus. So it's very funny because the female, she chooses a male to follow, and it is usually the dominant male, but not always, and she persistently follows this male, and she's very sticky, and in these days perhaps he will not let her to be around while he's cracking because she is really sticky, and then it. So what else should I say?
No, just to add that it's like Briseida said. So dominance is much more important for a capuchin male to be selected, and we don't have data to show that nut cracking is related to dominance acquisition in males. So I think we have enough data now, because we were interested in pursuing these ideas. I think it has nothing to do with sexual selection, at least in capuchins.
Yeah, I'm just remember some of the best crackers, if they were the dominant, and they were not. And we have really some crackers who were not reproducing at all. So.
So, is there any other question, comment? If there is none, thank you again very much for the symposium. Thank you everyone for your attention. And now we will have a break, a lunch break actually, so we'll go to eat. And don't forget that we have a special prize in ECA, especially for you who are students here. It will be for, I think, 25 reals, which is a special prize for this Congress, so use it, which is good. And then what's the other important information? Then we will actually start at 2 o'clock in the afternoon with the official opening and also with the keynote address. So don't forget to be here at 2 o'clock. And basically that's it. Thank you for your attention, and we will see you in the afternoon.
das perguntas. Ah, tá aqui.
Essa estava funcionando, essa é só ligar, mas agora eu vou te chamo.
Good afternoon, everyone. Sorry, I'm screaming. It's too loud. Good afternoon. Happy to see you here after lunch. And so we will start actually officially this meeting. It's already the 4th meeting in line of this Brazilians meeting on evolution of human behavior. There were already three others in different cities. This one is the first one that we are organizing over here. And many of you probably remember that this meeting is a little bit a time travel meeting because we have been organizing it for like around 3 years by now because we started before the pandemic and then everything was cancelled and then we restarted again. But luckily we are over here and probably some people are also watching us online. So all these talks actually will be recorded and they will be available freely. So we will have actually a nice database of presentations and talks and everything. So thank you again for coming. And I think that there is actually one important thing, more technical or, well, yeah, pleasure thing. Don't forget to come to the cocktail in the evening in the restaurant of Tres Pescadores, which is, everything should be actually in your emails and everywhere, so don't forget to get there, it will be nice. And we will have some space and time to socialize and discuss all the important topics about evolution and anything else. So, and now I will invite Edu Ottoni, who is my colleague from the Department of Experimental Psychology here at the Institute of Psychology, and he will tell a few words, important words actually, about his work and the evolutionary perspective in psychology. And then we are also looking forward to our opening keynote address. So, Edu, it's you.
Thank you, Jarka. So when they invite the old senior full professor, I guess they want more time travel than they already have. So let me tell you some storytelling. When I was about to start my undergrad students in the biology course, I had a friend. We were both amateur movie makers, but he became a professional one. So he decided not to become a biologist. Then he gave me his copy of Konrad Lorenz on aggression. And I was fascinated, but the word ethology was almost never mentioned in my undergrad years. And although there was important research on eusocial insects going on there, there was not a single discipline on animal behavior. By the end of my undergrad course, I was dealing with not so uncommon doubts about where to go on next, when a professor that was also a very good friend showed me a paper and suggested that, based on my interests, I should go have a talk with the author. And the paper was entitled Convite Justificativa para o Estudo Naturalístico do Comportamento Animal, which is an invitation and rationale for the naturalistic study of animal behavior. It was written by Professor Walter Hugo Cunha from our experimental psychology department, who has just passed away a couple of months ago. And I answered that invitation and was very well received in his courses. He was about to retire, but he introduced me to my future supervisor, his former student, Cesar Hades. Walter was raised in an experimental psychology department that was at the time constituted basically by behavior analysts in a strong behaviorist tradition, but he met the ethological approach and became its pioneer in Brazil. Our first generation of ethologists was thus constituted of psychologists, such as Cesar Hades, which was the greatest promoter of ethology in Brazil, who focused in non-human species, and Ana Almeida Carvalho, which was, I believe, our first human ethologist. So I was the first biologist to postgraduate in ethology in our department, but in even my generation, many biologists found a way there. And later disseminated ethology not only in the psychology bubble, but also back to biology courses. So one of my points here was to stress that even happening in a psychology department, our first movement towards an evolutionary approach to behavior was ethological. But later on, the evolutionary approach diversified, including relevant interactions with founding researchers of classical evolutionary psychology, such as Lida Cosmides and John Tooby, Marty Daly and Margot Wilson, but also with gene culture coevolution researchers like Kevin Leyland. I find our foundational story particularly important, actually invaluable, because some evolutionary psychologists at some point in history sometimes dismissed ethology as a somehow outdated approach to behavior. But I think that more recently there's an opposite trend, a clear opposite trend. And that's because the ethological thinking had a key contribution not only to the study of behavior, but I believe to evolutionary thinking as a whole. Because if you go back in time, Julian Huxley in 1942, he pointed three basic questions in the evolutionary study of behavior: adaptive function, evolutionary history, and immediate causation. But 21 years later, Nicole Tinbergen, the Nobel Prize in the ethological masterpiece, which is Aim and Methods of Ethology, he added the fourth question, development. Ontogenetic development was not missing in Huxley's list by chance, but because it was arguably an underrated process in the evolutionary model of the classic new synthesis. So nowadays, many authors state that Timberger's fourth question applies not exclusively to the study of behavior, but to the broader thinking of evolutionary biology as a whole. And all this to remind you guys, the newer generations in front of me today, of these lucky origins of our local ethological traditions, always close by to our evolutionary psychology endeavors, which help us never to forget this decisive fourth question. I see in the program that you already attended a short course and a symposium focused on development in this meeting. So I'm glad to see that we were very well prepared to the current debates on evolutionary theory, in great part due to our ethological legacy. So let's keep moving towards greater and greater consilience, which is the point of 21st one science of behavior, I guess. So I hope you enjoyed this meeting, and I'm looking forward also to watch the next lecture. So thank you.
Thank you very much, Edu. And I would invite Ed for the plenary talk, which we are very much looking forward, because we invited you. I already told him that he's among the Renaissance men who is studying basically everything about human behavior. So in case you are actually interested in, well, any aspect of human behavior, he's here to talk with you, to discuss. And we are looking forward to his talk. And now probably someone will need to help me. It's this one, right? Yeah. Yes. Got it. So this is actually working.
And then where's the which button? Is it?
It's only this one.
The forward button and the
Yes.
back button. Beautiful. Thank you.
Okay.
Well, thank you very much for inviting me. I'm delighted to be here. And today I'm going to basically just describe or explain evolutionary psychology, at least the way that I perceive it. Many of the points I make will probably be familiar to most of you, but I hope at least I might provide a few novel insights for some of you. And my own personal goal or journey was to try and understand, how do we understand human nature and human psychology and our emotions, our thoughts, as in a unified framework of all of nature. In other words, can we have a unified science, not just physics and chemistry on one hand and psychology and behavior on another, but can we all somehow view them as part of a single unified framework? And what I very quickly discovered is that, obviously I'm not the first person to think this, but this question goes back to the ancients. So we try and understand nature, the rocks, oceans, seas, clouds, hills, rivers, valleys, volcanoes, mountains, sand dunes, rainbows, mist, fire, water, drops, patterns. And to explain these sort of non-living kinds of things, scientists over literally thousands of years came up with abstractions, very powerful abstractions, things like objects that have a specific location in Cartesian coordinates, and that they have properties called mass and velocity and momentum and energy conservation. and experimental methods. And these have all been spectacularly successful in explaining the natural world, or many aspects of the natural world. Here's Galileo's famous experiment, rolling balls down an inclined plane to develop formal theories of motion. Newton, conservation of momentum, conservation of energy, decomposing white light into its component parts. These are all the things that we learn in elementary school and high school. But it seems like understanding organisms like ourselves all have these properties. We have mass, we have location, we can have velocity, we have momentum, conservation of energy. But it seems like organisms have a lot of properties that are not easily explained with these kinds of abstractions. Those objects in the foreground there with the fins on them somehow seem qualitatively different from everything in the background. Can we somehow come up with principles that explain both of these? And it's not obvious how to do that. If we look at these objects in the foreground that we call penguins, they have all kinds of strange properties that are all nearly identical. They all have a very complex structure, but they all share the same structure. It seems very opposite from the random sort of patterns that we see in the background there. And one thing that, as I will demonstrate in a moment here, that has been recognized for, again, centuries or millennia, is that organisms have parts that somehow seem useful or purposeful, things like teeth and ears and fur. But where does purpose or meaning, where does this notion of usefulness, how does that enter into a naturalistic theory of the universe? We have, if we look at our skeleton, it's this extremely precisely articulated set of elements, of parts that seem organized in a very specific way to achieve some purpose, like grabbing things. And if we look throughout our bodies, we see that the same kind of structure characterizes everything, not only in us, but in all organisms that we've examined. We have all these parts that are useful. They're highly organized, and within a species, they're nearly identical from individual to individual to individual. So the musculatory system, the respiratory system, and they're organized in systems, the circulatory system, the lymphatic system. And then, taking on the wise comments that we just heard, organisms develop. Again, a very strange and unique property that we start with a single seed and this tiny little physical structure by itself, autonomously, will develop over a relatively short amount of time into this incredibly complex, sophisticated structure that can then create more of these little seeds, that then create more of these complex physical structures. What kinds of principles can explain all this? And of course, this is true not just with acorns and oak trees, but here we have little penguin chicks, and they are going to very reliably develop into that adult organism. We need some kind of organizing principles, but principles that are not unique to living things, but that are part of a framework to understand everything. And here, of course, we see our own teeth. This is an X-ray of a child's teeth. We have the baby teeth that have already erupted. We have the adult teeth coming in behind them. Again, a highly stereotyped, happens in all of us, very reliably. And these things, these teeth, seem useful, purposeful. How do we get this notion of purpose or function into science? So how do we do this? How can life and non-life be understood within a single explanatory framework? So, of course, I'm not the first to think of this, nor were evolutionary biologists. If we go back in time to Plato, Plato was worrying about this and thinking about this. And he noticed, this is in one of his early works, the Protagoras. He has his dialogues here, notices that different creatures have different properties, and there's a logic to it. Those that have strength are not swift. Those that are weak do have swiftness. Some are armed, some are unarmed, some are devised, have other means of protection. So he's beginning to put together a theory that organisms have these parts that are useful. But where do they come from? Well, for Plato, these parts come from the gods. The explanation is, in some sense, a teleological explanation. So writing later in the Timaeus, Plato proposes that there's a divine craftsman, a demiurge. And of course, there are strong echoes here with later Christianity. And this divine craftsman is rational, he's purposive, he's beneficent, and he imposes order on chaos to produce a vast array of good effects. So just to give you a quick quote here, the demiurge created the sun, moon, and stars to mark time, for instance, which itself came into being as an image of eternity. He created eyes. So Plato's explanation, why do eyes exist? Well, they exist because the demiurge created them to the end that we might behold the courses of intelligence in the heavens and apply them to the courses of our own intelligence, which are akin to them, that we might imitate the absolutely unerring courses of God and regulate our own vagaries. So the explanation for eyes are that the demiurge, who's this wise, beneficial, rational agent, created them for a specific purpose. This external agent is creating order out of chaos. So that's where the purposefulness and the utility comes from, at least according to Plato. However, Plato was not the only thinker grappling with these problems. There were the atomists, Democritus being a key example. And they rejected this teleological explanation. For them, the universe is explained by movements and interactions of a small number of particles, atoms, and they interact. And they had many ingenious arguments for how such a small number of particles and a limited number of interactions could explain everything we see in the world. And critically, there's no demiurge. Causation is only due to blind chance and necessity. This is very similar to our modern day physics. But it was very difficult in this framework to get to purpose and organization and function that we see in living beings. So Aristotle was grappling with both. He was grappling with the atomists and their arguments, but those arguments really didn't explain living things very well. And with Plato, who can explain living things as the product of a wise demiurge, but is bringing in this external being to kind of explain order, this external intelligence. So to explain some phenomena, Aristotle did invoke atomist arguments. Rain, for example, occurs of necessity and itself has necessary and chance effects. So why should not nature work? Not for the sake of something, nor because it is better so, but just as the sky rains, not in order to make corn grow. There's no purpose here, but of necessity. What is drawn up must cool, and what has been cooled must become water and descend. And the result of this is that corn grows. Similarly, if a man's crop is spoiled on the threshing floor, the rain did not fall for the sake of this, in order that the crop might be spoiled, but that result just followed. So for Aristotle, many things in nature are just chance and necessity, much as we might see in modern physics today, explaining much of the natural world. But Aristotle we needed an explanation for animals and the structure and purpose that we see. They cannot be explained only by blind chance or necessity. So teeth, for instance, are admirably constructed for their general office, the front ones being sharp so as to cut the food into bits, and the hinder ones broad and flat so as to grind it to a pulp. There's purpose here, there's function, there's organization. And because the teeth grow in this way in all humans, Aristotle goes on to argue, this pattern cannot be due to chance, contrary to the atomists. Instead, Aristotle concludes that there has to be what he called a final cause. Somehow immanent in the organism is some causation, some causal force that forces the organism to reliably develop teeth in every single member of that species. But there's no demiurge. There's no wise, beneficial creator. So Aristotle does have a teleological argument, but unlike Plato's, it's local, not global, and it's immanent in the organism, not external to it. And that's critical. And he wrote a whole book on this. So something is not just a an aside. It was really a central focus of his scientific framework. Now, Galen, famous Greek physician living in Rome, he was grappling with both Plato and Aristotle, so he accepted Plato's explanation that there is a divine craftsman. But unlike Plato, Galen also adopted many elements of Aristotelian thinking and understood that the structure of organisms are for the benefit of the organism itself. So unlike Plato, where our eyes are constructed by the demiurge so that we can observe the heavens, for Galen, who did many, many dissections, so he was very much an empiricist, and he did hundreds, if not thousands, of dissections of pigs and monkeys to try and understand the structure of those organisms, using empirical evidence on the structure of pigs and monkeys, and assuming that they were very similar to humans, trying to understand how that structure benefited the animal itself. And he understood those interrelations of parts within the organism in materialistic terms, not teleological terms. And Galen's work was so influential, it really held sway, at least in Europe, for 1,400 years. So jumping over lots of history here, we get to William Harvey, who begins to understand all of these parts that Galen was grappling with in mechanical terms. So Harvey writing about the heart, the chief function of the heart is the transmission and pumping of the blood through the arteries to the extremities of the body. So Harvey is combining the increasingly sophisticated mechanical views of his age, but retaining the teleological aspects that these things are there for a specific purpose. Now jumping ahead again, I'm over a lot of history here, to William Paley. William Paley wrote a very influential book called Natural Theology, and he is increasingly developing this mechanical view of organisms. And so for Paley, there's still a divine creator. Of course, now it's the Christian God. But he's adding two things. Organisms are machines. They're not just like machines. Organisms are machines. They have all these parts. These parts have very specific relationships to each other. These parts benefit the organism, but they're also adapted to their environment. So the bodies of animals hold in their constitution and properties a close and important relation to nature's altogether external to their own, to inanimate substances, and to the specific qualities of these. They hold they hold a strict relation to the elements by which they are surrounded. Can it be doubted whether the wings of birds bear a relation to air and the fins of fish to water? They are instruments of motion, severally suited to the properties of the medium in which the motion is to be performed, which properties are different. Was not this difference contemplated when the instruments were differently constructed? So Paley is giving us a model, a conceptual model of organisms as machines that are adapted to their environments. And of course, here are birds clearly adapted to flying, and fish clearly adapted to swimming. And it was this model that Darwin read. As a young man, before he became a naturalist, he was assigned William Paley in class. And here's Darwin writing in his autobiography, crediting Paley. So just to quote here again, in order to pass the BA examination, and these are Darwin's words, it was also necessary to get up Paley's evidences of Christianity and his moral philosophy. The logic of this book, and as I may add, his natural theology, gave me as much delight as did Euclid. The careful study of these works, without attempting to learn any part by rote, was the only part of the academical course which, as I then felt and as I still believe, was of the least use to me in the education of my mind. I did not at that time trouble myself about Paley's premises, a God that did all this, and taking these on trust, I was charmed and convinced of the long line of argumentation. So in some sense, Paley has given Darwin the question that Darwin will then answer. Where do these machines that are adapted to their environments, how do we explain that, not in supernatural terms, but in naturalistic terms? And of course, Many years later, here is Darwin writing in The Origin of Species. He gives us his theory of natural selection. We get organism design without a designer, without final causation. So just to quickly quote here, it is preposterous to attribute to mere external conditions the structure, for instance, of the woodpecker with his feet, tail, beak, and tongue, so admirably adapted to catch insects under the bark of trees. In the case of mistletoe, which draws its nourishment from certain trees, which has seeds that must be transported by certain birds, and which has flowers with separate sexes, absolutely requiring the agency of certain insects to bring pollen from one flower to the other, it is equally preposterous to account for the structure of this parasite with its relations to several distinct organic beings by the effect of external conditions or of habit or of the volition of the plant itself. We need some other explanation, and of course the explanation that Darwin gives us is natural selection, heritable variation, and differential reproduction. We finally now have a naturalistic explanation for design without a designer, for organism structure, all of the stuff that philosophers and naturalists had been struggling with literally for millennia. We now have at least the beginnings of a naturalistic explanation. Now I'm jumping again here to Erwin Schrodinger. He's the famous physicist who gave us quantum mechanics. And he wrote a book that those of you who are interested in evolutionary approaches to organisms, I strongly recommend you read. It's called What is Life? It was written in 1944. It's actually a series of lectures that Schrodinger gave that were eventually published in a book. And this is before the discovery of the structure of DNA. So Schrodinger is trying to reason, how does the heritable variation and this heritable structure that is essential to the theory of natural selection, how can physics explain this? How is that consistent with what we know from physics? And so Schrodinger is starting with the problem: the unfolding of events in the life cycle of an organism exhibits an admirable regularity and orderliness, unrivaled by anything we meet in inanimate matter. How is this possible? And Schrodinger gives us two principles from physics, how you can have order in what is essentially, if we look at it at the atomic level, a very highly chaotic and disordered universe. With thermodynamics and entropy and heat death, how do we get orderliness? And there's two ways, according to Schrodinger. One is statistical mechanics. This is order from disorder. So macroscopic structures, if you looked at them at the microscopic level, would be highly chaotic. But those highly chaotic motions of atoms would basically average out, and we can get stable physical structures like chairs and organisms. And so the organism, as a whole, must have a comparatively gross structure in order to enjoy the benefit of fairly accurate laws, both for its internal life and for its interplay with the external world. So we, as organisms, can have a stable structure, literally because we're so big. You could not build an organism from atoms. It would be too chaotic and random. You could only build organisms macroscopically, where we can take advantage of statistical mechanics. But that doesn't work for heritability, because from heritability we have a tiny fertilized egg that somehow contains everything it needs to develop from an acorn into an oak tree, or from a fertilized egg into a human infant. And so Schrodinger was struggling with this problem. What in physics allows that to happen? And he makes an argument in the book. I'll just give you the quick quote here. We can get order from order on the basis of quantum theory. And the basic thing is, I won't go over the argument here, but he shows how quantum mechanics can give us a stable molecule. And so he reasons that the substance, what he calls the aperiodic crystal, has to be a molecule that carries the hereditary information. Again, this is before the elucidation. This is before Watson and Crick. So just to quickly read here, it has often been asked how this tiny speck of material, the nucleus of a fertilized egg, could contain an elaborate code script involving all the future development of the organism. A well-ordered association of atoms, atoms endowed with sufficient resistivity to keep its order permanently appears to be the only conceivable material structure that offers a variety of possible isomeric arrangements, sufficiently large to embody a complicated system of determinations within a small spatial boundary. Indeed, the number of atoms in such a structure need not be very large to produce an almost unlimited number of possible arrangements. So here Schrodinger is reasoning a priori, there's got to be a molecular code somehow inside the fertilized egg. Consequently, we may safely assert that there's no alternative to the molecular explanation of the hereditary substance. The physical aspect leaves no other possibility to account for itself and of its permanence. It is these chromosomes that contain some kind of code script, the entire pattern of the individual's future development and its functioning in the mature state. Every complete set of chromosomes contains the full code. So that code, of course, about a decade later, was now firmly established to be the molecule DNA. In humans, it contains 3 billion nucleotides. These are the molecular or the atomic units or the molecular units that Schrodinger was talking about. We all know them as the A's, the C's, the T's, and the G's. There's 4. So Schrodinger reasoned that there didn't need to be very many of these. In fact, there weren't. There were just 4. And there are a sequence of these, 3 billion of them. And we now know that about 1.5% of those specify proteins. That's about 20,000 genes, 1 gene per protein. So everything we're made of, every neuron in our brains, every cell in our bodies, are made of these proteins that are specified in this 1.5% of the DNA. We know another 10% of the DNA is non-coding. It somehow regulates, I'll talk a lot more about this, somehow regulates the expression of those proteins. We're still working that out. There's massive amounts of research on that right now. And it's also, it seems likely there's still debate on this, but maybe 85 to 90% of the genome might just be junk. It might really not contain have any real impact on the phenotype whatsoever. That's important too. And that's important for this. We have, I think, kind of in popular discourse, and also if I looked at my biological anthropology textbooks, there's 2 conceptual models of how we should think about DNA, and the most popular one, the most common one, the one that's taught in most textbooks, I think, is what I'm going to call the genetic difference model. I want us to reject that model, or not fully reject it. It's actually correct, but it's trivial, or compared to what I want to promote now, what I call the genetic program model, which is profoundly, this is the code script that Schrodinger is talking about. It has profound biological significance, and it's quite different from the genetic difference model, the one that unfortunately is more popular. So quickly, what do I mean by the genetic difference model? Well, this is the model where we look at genetic differences between individuals, and we correlate those genetic differences with differences in behavior, or differences in skin color, or differences in metabolic activity. And we do that between individuals, and we do it between populations. And this is kind of the basis for all of the genetic kits that many of us have used in the United States, 23andMe, Ancestry.com. I'm not sure what we have here in Brazil, but they tell us where we had some ancestors here, and we have some ancestors there, and we have maybe this risk factor for this disease, and this risk factor for that disease. And these are all based on genetic differences between 2 individuals. And again, it is true. There are genetic differences. Here they are. Here's a large study from the 1,000 Genomes Project Consortium. Each little tiny cross here, basically on the vertical axis, we have the number of differences between a specific genome and what's called the reference human genome. So there's a reference human genome, and then you sequence somebody else's genome, and you count up how many differences are there between this individual's genome and the reference human genome. And what you can see on the vertical axis, 3.8, 4, 4.2, those are millions of differences. And here we have a large number of individuals from the 1,000 Genome Consortium in different populations, Great Britain, Asia, African populations, Japan and so forth, Finland, Central Europe. And what you can see is that all of these various individuals have between 4 and 5 million genetic variants compared to the reference human genome. And I actually got a deal where I could get my whole genome, the whole genome sequence. I did that, and I got a file back, and the file had 5 million lines, and the 5 million lines are every point where my genome differs from the reference genome. And that sounds like a lot. But remember, there are 3 billion nucleotides in the entire genome. So 4 to 5 million variants is less than 1%. It's a tiny, tiny fraction of the entire genome. Most of our genome does not differ. And that's the part that is the most important. So let me just give you a diagram kind of showing where those genetic differences are. So in this, let's see if I can use my pointer here without screwing everything up. So these columns here, this is Africa, Europe, Southeast Asia, East Asia, and the Americas. These are the total number of, so here's the entire 3 billion nucleotides, 2.9, but let's say 3 billion nucleotides in the genome. Here is the percentage of the genome that differs across all of these different populations. So these are single nucleotide variants. So this whole thing is a zoom in on this little tiny fraction of the genome here. And this is the total number of differences, 91 million differences, not in a single genome, but across a large number of genomes. And then it's saying, let's organize those differences. Where do we find them? And so this whole chart is going to be from 0 to 100% of all of these 91 million differences. So these are variant counts. So 14 million of them, or almost 15 million here, 16%, are found in Africa, and they are rare variants. Now, what does that mean? That means that all these variants are almost only found in Africa. So you might think, hey, man, Africans have 15 million, almost 15 million variants that are not found in any other population. But they're rare. What does that mean? That means most Africans don't have those variants. These are basically new mutations that mostly that have very minor, probably negative phenotypic effects, if they have any phenotypic effects whatsoever, and they haven't been selected out. So the main message of this chart is that most genetic variants between individuals are rare, very rare, and they're population specific. And we can basically understand them as relatively recent mutations that probably have not yet been selected out of the genome. And only a tiny fraction of the total number of variants are common variants that we might find if we took any 2 individuals, that one of them would probably have these variants. There's only about 4% of the total variation in the human genome is actually common genetic variants. Most of it's rare. So yes, it's specific to Africa, it's specific to Southeast Asia or Europe, but even 2 individuals from Africa would not likely share those variants. Okay. So this is why I want to reject this model as being biologically significant. It's really not. Most of this genetic variation between individuals is noise. It's not the code script that determines the structure of the organism. That is the job of the genetic program model, and that's what I really want to emphasize here. Now I'm going to try, I have to do something a little tricky here to make this work. I had to do this to get the animation to stop. And let's see, I can now stop it. There we go. Okay. Many times when it comes to the word program, when people hear the word genetic program, they think of something that's very fixed and static. And so what I want to offer you is a little mathematical formula that is fixed. Let's see, where's my pointer here? This is the formula. This is the algorithm. This is the program that's running right here that we'll see in a second. Notice how simple this program is. This is called a logistic map. You can Google it. There's some really cool videos on this on YouTube. But it's super simple, and it's basically a model of population growth. X is population size. And we're basically saying that population size at time n plus 1 is equal to the population size at time n times a parameter, the growth rate, lambda here. So to get the population at n plus 1, you take the population at times n, multiply it by lambda, and you get n plus 1. But there's the second term here, and this is also a very simple term, that when the population starts off at 0, this just equals 1, and you just get the population growth rate here as we laid out. But as the population size gets close to 1, which is 100% of its maximum size, then this term is going to get smaller and smaller and smaller, and so you basically multiply the growth of the population by a term that represents that it's basically getting close to maximum, so that's going to decrease the growth rate as the population approaches its maximum. Now, what's interesting about this? On the vertical axis, we have the value of x. And then on the horizontal axis here, we have n, 1, 10, 15, 20, da-da-da-da-da. So this is the growth of the population over time. And what you're going to see is that this extremely simple program generates incredibly complex behaviors or outputs. So I'm going to play it here, and we'll watch it cycle through a couple times, if I can figure out what I'm doing. So it's already started a little bit, but maybe if I can, I'm afraid I'm going to screw something up. Let me try it from the beginning. Okay, I'm going to do this. I'm going to do that. Okay, now it's running. And what we're doing, I'll start it again. There's a little, what we're going to do is we're going to run it for different values of lambda, different values of the growth rate. So low values of the growth rate that are less than 1 are going to lead to the population to decline and go to stink. And values of lambda that are greater than 1 are going to lead to population growth. And what we're going to watch is an animation of what the population trajectory looks like for different values of lambda. So let's watch it. So as we can see here, lambda is less than 1, the population goes extinct. Now lambda is increasing, population is growing. But now watch what happens when lambda, this term here, gets over 3. We start to get incredibly chaotic, or variable behavior, not chaotic yet. But as soon as we get over here, it goes nuts. And so the lesson that I want to draw from this is not that this particular formula is of relevance to what I'm talking about, but just the general principle that very simple programs can generate enormously complex behavior. And what I'm going to kind of ask you to keep in mind is that this is a fixed program, but I want you to kind of think of lambda as an environmental input to the fixed program, and that'll become relevant in a minute here. Now let me get this going again here now. Oops, let's see, how do I start the slideshow? I'm clicking the wrong button, I think. Is that the button? Thank you. Okay. Oop, we got to get rid of that. It's gone, great. Okay. So now turning to development. We have a single fertilized egg. We're now talking about humans. And over the next 9 months, it's going to divide and divide and divide and divide and go from a single cell 9 months later to an infant with 2 trillion cells, all different. And then over the next 18 years into an adult of 30 trillion cells. And these cells are all different kinds of types of cells. So a single cell now becomes a multitude of cells, a huge multitude of 30 trillion different cells. Many of them are red blood cells, muscle cells, neurons, hepatocytes, bone marrow cells, lymphocytes, epidermal cells, and so forth and so on. So how does a molecule in this single fertilized egg determine this universal developmental sequence? And we're not going to go through in many details, but what I want to offer you for the genetic program model is that what's important about the genome are not differences between the genomes of individuals, but the interactions within the genome, within a universal human genome. So how does the genome interact with itself? Well, there's many ways, but the most important way is with a particular kind of protein or gene called a transcription factor. And these are genes that regulate other genes within the genome. And the basic way they work is that you have a gene that produces a protein called a transcription factor. But this transcription factor has a special, what's called DNA binding domain. It's basically a key. It's a special, highly specific, and each transcription factor has a different one of these different keys, but it has a very highly specific pattern that binds to very specific regions in the DNA. And by binding, by these transcription factors, which are produced by some genes, that then bind to specific regions of other genes, they regulate the activity of those other genes, either upregulating their expression or downregulating their expression. And these transcription factors can also have parts of them, they are proteins that bind to ligands, or ligands bind to them, that then alter their regulatory behavior. So the genome we should think of it as an incredibly complex machine in its own right, comprising parts of the genome that are interacting in extraordinarily complex ways with other parts of the genome to achieve very specific effects. And this is just a very simple example of one of these. These are called gene regulatory networks. And these are how we get all the different cell types during development. So here are some transcription factor genes that produce transcription factors that then bind to different genes that then produce more transcription factors. You get a positive feedback loop here. And so then you get this positive feedback loop of producing proteins that will cause particular cells to develop into neurons, others to develop into muscle cells, others to develop into blood cells or lymphocytes or what have you. So there's a large number of networks defined within the genome that determine cell identity. Here's just a couple examples for interneurons and motor neurons. And we still don't understand these very well. These are still very poorly understood, but they are a focus of a massive amount of research right now. Now, to really understand these things, like we have to do with any physical system, you have to basically be able to manipulate the system. And we can't really do that ethically with human fetuses because you can't take a developing baby and say, oh, we're going to knock out this gene and see what it does. No. So we do it with model organisms. Sea urchins are very common. There's also mouse models. Here's a sea urchin developing from the early stages of 4 cells across time here. And this is by knocking out different genes. This is the circuitry, the gene regulatory network that was inferred that's guiding the development of the early sea urchin. And you can see how complex it is. And this is just part of the gene regulatory network or the genetic program that's guiding the development of the sea urchin. Same thing's going on in principle in humans. Now, a critical thing is that these gene networks can detect and respond to the environment. So here's just a very quick example. In our immune system, this is called a toll-like receptor. This is a protein that goes through the cell wall here, and it binds to different parts of bacteria, detecting infections from bacteria. Triggers a signaling cascade here, producing transcription factors that then alter the behavior of immune system cells to respond to a bacterial infection, for example. So a critical thing about these gene regulatory networks, they're not operating blind. They are like that logistic map, where lambda we can think of as the environmental factor, that changing lambda, changing these environmental factors, alters the behavior of the gene regulatory network, often in profound ways. Now here's an example. These are water fleas. These 2 water fleas are genetic clones. They're genetically identical. But what you can see is that they're morphologically quite different. The one on the right here, or the one on the left here, has this very marked helmet that's missing on the one on the right. And this huge pointy tail, much different than what we see on the right here. So what's going on with these water fleas? Well, they live in a marine aquatic environment, and they are predated upon by fish. And building this helmet is very expensive. It takes a long time, a lot of resources. You don't want to build the helmet if there's no fish, if there's no predators around. But if there are predators, you've got to build that helmet early on. You can't just develop it at the drop of a hat. You can't say, oh, there's a predator, I need a helmet. You need to do that early on in development. So water fleas have a gene regulatory network that can detect chemical cues of predators. If those chemical cues are absent, no helmet develops. But if the chemical cues of the predator are present, the water flea develops this defensive helmet. Now you might ask, is there anything like this in humans? And I'm going to argue we have many examples in the room here. We have males and females. And males and females are basically created by the same genetic program, but that just gets altered due to a genetic switch. So here's sex determination in mammals, including humans. You get the initial development of the genital ridge here. If you happen to have, now we all think, hey, I've got a Y chromosome that has all the genes to code for male. If I don't have it, I develop into a female. That's actually not how it works. There's only a single gene on the Y chromosome that determines if you're going to be a male or not, and that's called the SRY. And if you have it, it triggers a gene regulatory network on chromosome 17. It basically binds to what's called SOX9 here and develops a whole regulatory cascade that leads to testicular development, And if you're lacking SRY, you then go down a different regulatory path and get ovarian development. But critically, all of this are on the autosomes. They're on the genes that are shared by both males and females. And this has been known for well over 100 years, that basically our genomes contain all of the code to create a male or a female, and it just depends on the presence or absence of this genetic switch, SRY, not on the other genes on the Y chromosome, of which there are actually very, very few. And I want to just offer this as possibly, we need this in our conceptual toolkit. We know about the water fleas, we know about sex determination in humans, but we might have other things like that detect environmental cues and trigger different developmental trajectories. So many of you probably know about fast and slow life histories. Could this be due to some environmental switch? Or maybe even a genetic switch? Possibly. We actually have evidence in our immune system that our immune systems develop differently if we're in very rural kinds of environments versus more urban environments, for example. So this is just an idea that we should keep in mind, that there might be different developmental pathways beyond just sex or beyond just the immune system that are sensitive to environmental cues early in development. Now, these gene regulatory networks guiding development, they're not just in us humans, of course, they're very conserved. Conserved means they don't really change genetically, even between mice and humans, or across all mammals. So these gene regulatory networks, we can learn, are very, very conserved. They don't change, and that means within humans, they're essentially identical. So our basic model is we have this whole genome. These are the green squares. There's a tiny amount of genetic differences. That's mostly noise. And then different kinds of inputs to these gene regulatory networks, SRY, genetic switches, epigenetic switches, environmental conditions, trigger the program to cause it to behave in different ways to develop a phenotype that is tuned to local environmental conditions. In the water flea case, are there predators or not? In the human case, who knows? We know maybe in our immune systems, are there lots of pathogens or not? We might need different phenotypes to deal with those. Okay, how do we bring this back to now psychology? Everything I've said applies to our entire phenotype, not just our psychology. How do we bring this to psychology? How do we have a machine model, like a pump of the heart? What's the analogy or the analogous sort of mechanism or machine for the brain? And of course, as we all know, it's the computer. So the computer is our model of the machine that is the brain, that is cognition, that is psychology. And the different parts of our brain are not levers and pumps and valves, but instead what we might think of as apps. Just as your smartphone is a single computer but has a lot of different apps to do all kinds of different things, you need different apps for taking pictures, for writing notes, for talking on the phone, for sending messages. The argument here is that we need different apps in our brain to deal with all the different information processing challenges that humans faced over the course of their evolution. So this is evolutionary psychology, that we have multiple gene regulatory networks that evolved by natural selection to generate tuned psychological adaptations, adaptations that are tuned to our local environment, apps, algorithms, modules, pick your term. These solved important problems of survival and reproduction posed by, and for things to evolve by natural selection, it takes a long, long time. So it had to be problems that were recurring over lengthy periods of the ancestral environment. So what is this environment? Well, here we have a plot of fossils, human fossils, or fossils of human ancestors, I should say. And I'm kind of missing a little element here, but we'll make do. On the y-axis, so each dot here is a fossil. So Lucy is down in here somewhere. Well, we don't have Neanderthals in this particular plot, but they would be up in here somewhere. Homo erectus down in here. These are millions of years before the present. So this is today, 1 million years ago, 2 million years ago, 3 million years ago. And what we have on the vertical axis is the brain size of these fossils. And this is the normal the modern chimpanzee range is right about here. And the modern human range is right around here. And what we can see is about 3 million years ago in the Pliocene, our ancestors' brains were about the size of chimpanzees. And then over the next 2.5 million years, starting right about here, this is the beginning of the Pleistocene right here, and going over this 2.5 million year period, our brain size triples. And where is that happening? It's happening in Pleistocene Africa. So the major structures of human cognition are evolving in this environment in the Ice Age, in the Pleistocene, right here, as you can clearly see in this plot. So, can we know anything about that past? Well, yes, we can. So here's just some basic climatic data. On the x-axis, again, we have time. Here's today. On the vertical axis, we have global average temperature. And this solid horizontal line here is the average temperature today. So values above this are warmer, values below this are cooler. Here we have the time of the common ancestor of all primates. You can see the environment was very warm and wet, maybe plus 12 degrees back then. And then the climate undergoes a long period of cooling and drying over this long period. So here, about 6, 7, 8 million years ago, we have the common ancestor of human and chimps. And here's where we have those fossils I just showed you, starting right around here. And the first members of our genus Homo show up right about here, 2.5 million years ago. And this is when brain size begins to increase. And what you can see is that the climate is, we're entering the Ice Age. And not only is it cooling, but over this period when human brain size triples, it becomes extraordinarily variable. And many people, myself included, think this climate variability has a big part to do with that increase in brain size. And modern humans show up in Africa right around here, 200,000 to 300,000 years ago. And then we get the end of the Ice Age, and here we are today. Now that's just temperature. But how strange was this Pleistocene African environment? Well, it really wasn't that strange. Here's just an artist's depiction of all the animals and landscape that we would have seen in Ice Age Africa. And we can see it looks extraordinarily familiar to what we would see today in any kind of savanna environment. There's animals, there's plants, there's birds, there's large mammals, there's small ones, there's water, there's rivers and lakes and ponds, there's clouds, there's rain, there's hills, there's forests, there's grasslands. It's an environment that's really almost exactly what we see today. It's not that exotic. So modern environments are reasonable proxies for the Pleistocene environments. And the kinds of environmental features that were relevant to human survival today were relevant then. Physics, mass, gravity, light, sound, motion, fire, weather, temperature, rainfall, wind, geography, lakes, rivers, resource locations, infectious diseases, people, faces. There were faces, there were infants, there were children, there were adults, there were males, there were females, there were kin, there were non-kin. There were groups, there were animals, there were arthropods, there were parasites, there were tetrapods, birds, mammals, predators, prey. And there were plants that had leaves and fruits and seeds and toxins. So all the kinds of things that are critical to survival and reproduction today have been critical to survival and reproduction for a long, long time. I keep going to the keyboard here. I don't need to do that. Got the button. Now, of course, many people would say, what about learning? We can learn about all these things. Yes, we can and we do. But learning is not simple. And this is just a classic model of Pavlovian conditioning. And I just throw it up here to show you. Learning is complicated. It's a complicated system to be able to learn useful information from the environment. That requires specialized psychological adaptations to do the learning. And the same goes for culture. We need specialized adaptations to learn culture, use culture, and transmit culture to others. A blank slate doesn't learn, a blank slate doesn't learn culture. There needs to be cognitive structure to do those things. So let me just give you some examples of how we can apply these principles to begin to explore new areas of evolved human psychology. This is some great work by a colleague of mine, Annie Wirtz. And the basic adaptive problem is that plants are important sources of food, but many are highly toxic and poisonous. So how do we deal with that problem? So in experiments that Annie has done, she wondered, how early in development do we begin to recognize that plants might be dangerous? So she works with young infants in this kind of paradigm, and it's a very simple thing. She takes these young infants and she puts in front of them different kinds of objects. So some of those objects are natural objects like rocks and seashells. Others are artifacts like trowels and spoons and keys and hairbrushes. Others are novel artifacts that the kids would never have seen before. Some are artificial plants, and some are real plants. And then she simply says, how long does it take for the infant to reach out and touch the object? And what you can see here, these are the different kinds of objects here. So here we have natural objects, familiar objects, novel objects, artificial plants, real plants. And then this is the delay of the infant to reach out and touch the object. And what you can see is they relatively quickly touch these things, but they take a lot longer to touch these things. In fact, they won't usually even touch them. They look to the adult to see if it's okay. So this is at least one piece of evidence that infants are more cautious around plants than other kinds of objects, and the explanation might be that they have an innate knowledge that plants can be dangerous, that could kill them. Okay, here's a second example, remembering locations of food. Where is the food? You've got to have specialized memory to remember where food is. So in this experiment, they took individuals into a farmer's market that had a number of different stalls with different foods, things like lettuce and cucumbers and cherries and oranges and avocados and honey. And they take them through the market. Each student, or each participant, I should say, goes a different path led by a researcher, and they taste a food at each stall. And then after that, they are taken into the middle of the market where there's a big dial, and they have to kind of point. They say, okay, where was the lettuce? Rotate the dial and show me where the lettuce was. Okay, tell me where the blueberries were. Rotate the dial and show me where the blueberries were. And so forth and so for all the foods that they've tasted. And then what we have here is the error. The pointing error. How far off are they? So 0 is no error. They exactly locate the direction of the food stall with the food. So this is the error. And then on this axis, on the x-axis, is how nutrient dense is the food. And what you can see is there's a very interesting pattern. People are pretty inaccurate with foods that have low caloric content, lettuce, cucumber, zucchinis. But they're quite accurate with foods that have high caloric content, honey, almonds, olive oil. So this is at least some evidence that we might prioritize memorizing the locations of nutrient-dense foods over those that are much less, have far fewer nutrients. Okay, third example. Animals are also an important source of food, but they can be dangerous. So in this study, it basically does a study where you see if you can detect a change in a scene and how quickly you can detect it. So there's a mask, and then you see a scene for just a short amount of time, a quarter of a second, and then there's another mask, and then you see the same scene, but something has changed in the scene. And the question is, how long does it take people to detect what has changed in the scene? So using Photoshop, the researchers just changed a tiny little element of the scene. Well, what did they change? Here's some examples. And what they change is the presence or absence of an animal or a vehicle or a person or a bird. Here's a little bird here. Here's a person here. And they basically Photoshop, or a cup here, and they Photoshop these things out. So you're seeing these pictures flipping back and forth, and you have to say, okay, something has changed, what is it? And what they find is, and then how accurate are they? Sometimes they never detect it. And what they find is people are very quick and very accurate at detecting that what has been removed is either a person or an animal. And they're much less good or accurate at picking out even things like vehicles or buildings, like in this one. They took out this whole building, and people were very slow to detect that an entire building had disappeared. So the argument here is that we are specialized to detect people and animals when they appear on the scene because they are opportunities but also threats. Okay, final example, mate choice. This, of course, is what evolutionary psychology is notorious for, sex. Finding a mate, we don't just mate randomly. Nobody does. We don't just close our eyes and pick the first person of the opposite sex and have sex with them, or the first person, or the first object. We usually put a huge amount of time into picking mates. And there are some qualities that are universal, that there's no predicted sex difference. So this is a very large, and there are other properties where we do predict a sex difference. And this is a large study, it's actually a replication study across 45 countries for mate preferences. And what we have here, and each dot here is one country. And what we have here is the sex difference. So this would be, it's males minus females. So if the female scores are bigger, we're shifted over to the left. If males are bigger, we're shifted over to the right. And for the properties that are predicted to be important for both sexes, like health and kindness and intelligence, we see a little bit of a sex difference, but not much. This is where we have no predicted sex differences, and indeed the sex differences are quite small. Males and females are very, very similar in preferring mates that are healthy and intelligent and kind. But we do have some predicted sex differences. We predict that females are going to prioritize, so both sexes want resources, both sexes want fertility, but due to pregnancy, females are predicted to put a greater emphasis on resources and males on fertility. And this is what we see, good financial prospects. There's a female bias, a very clear female bias. So both sexes like this, females like it more. Physical attractiveness, which is thought to impact fertility, both sexes like this, males like it more. And then age choice, which is both a combination of fertility and resources. There's a very consistent that females prefer mates that are a few years older, not that much, but 2, 3, 4 years older than them, and males prefer mates that are a few years younger than them. The argument being that younger females have higher fertility and older males have more ability to produce resources. So we see, again, some at least evidence for human universal mate preferences here and sex differences in the predicted direction. But these sex differences tune to the local environment. So this is, again, the same data. Each dot here is one country. But the countries are now separated out on the x-axis here by the gender equality of the country. And we can see that in very gender unequal countries, the sex difference in age, or the sex difference in age preferences is quite large. But as gender equality increases, the sex difference decreases. So as the differences between the sexes decrease, the preferences for an age difference also decreases, suggesting at least some evidence that mate preferences are tuned to the local social environment in this case. Now, hopefully it doesn't need to be said, but I'll say it anyway. We've got to avoid the naturalistic fallacy. just because nature is one way, just because human nature is this way or that way, doesn't mean human nature should be that way, that it's good to be that way. Aggression exists, jealousy exists, all kinds of bad things are probably part of human nature, but we are very justified in trying to reduce those. This is from Hume, and then from Moore, very similar thing. Just because something is natural doesn't mean it's good. Okay, to conclude. Organisms comprise parts that were designed or engineered by natural selection to promote survival and reproduction in the EEA, the environment of evolutionary adaptiveness. And the brain, so that's the organisms in general, and the brain is the same way, no difference. It has parts, psychological adaptations, that were designed or engineered to promote survival and reproduction in the EEA. And these parts are generated and tuned by universal gene regulatory networks that take environmental inputs. So remember, what is fixed in evolutionary psychology are these gene regulatory networks, not even the psychological mechanisms, and definitely not behaviors. Remember that logistic map. The formula is fixed, but the lambda can change, and that can dramatically change the behavior. So we became human in the African Pleistocene, we all did. It posed many adaptive challenges. And that gives us these universal gene regulatory networks that create the phenotypes that we all have today. Thank you very much. Have I left any time for questions?
Thank you very much for a wonderful talk. There is really lots of food for thought. I think that I will need to watch this talk again, like at least 3 times to digest everything. But are there any questions right now? Oh, and if you want to, you can also, the local people can ask in Portuguese if you want to. We can try to translate. You don't need to be afraid asking in English, if that would be a problem.
Ed, thanks for your presentation. Just a few minutes before your presentation, Ottoni talked about kind of consilience between different approaches to evolution of human behavior. Do you think that, for example, the perspective of evolutionary psychology of a reliable development and recognizing this difference between other approaches on the evolution of human behavior which not work with a reliable development, so acknowledging these differences can lead to this consilience, for example?
Yeah, that's a great question. First of all, I would say all these approaches are generating data. And I would say we're probably all wrong. Evolutionary psychology is probably wrong, ethology is but we're all right in a certain way. So that the data, the evidence generated by all these different approaches are going to have to be reconciled in some framework. I would also say evolutionary psychology grew out of ethology, and it grew out of a lot of these other traditions. So I would say some of that consilience is already happening here. And I guess the third thing I would say is empirical tests. We have to really, we kind of come up with these grand frameworks, but at least my own experience with my own projects is I get this great idea and then the data don't support my idea. So I'm very much, very strongly in support of robust empirical programs that are really focused on testing ideas. And in your studies, I encourage everyone, when you put together a study, don't just test your idea from evolutionary psychology or from ethology, but take different hypotheses from the different traditions into the same study and test them against each other. And that way is how I think we will achieve the consilience that Eduardo is looking for in our experimental approaches and our empirical approaches, that we don't just look at our own approach, but we take the hypotheses from the other approaches, put them in the same study, and pit them against each other head to head. And what I often find, at least in my own studies, is nobody's right, and we have to kind of come up with a new theory. So I would hope, yes, that we are all steadily marching toward a conciliant framework that will probably look a little bit different from anything we see today, would be my guess, but we'll see. Thank you. Yeah.
That's a very positive view, which I would like to share. Other questions, comments, suggestions?
So I'm very new to all of this. Just about the sex thing, like 2 slides ago, something like that. Yes. Does it take in account like the patriarchy? Like men are like being told over and over, like, yes, like young girls to prefer, like the socialization interferes in the results? Or it's just like, oh no, it's just biology?
So in this study, each dot here is a different country. So there's 45 different countries here. And you're right, what we see here is in this chart, each country has a gender equality score, which would be, in some sense, a measure of the patriarchy. So highly patriarchal cultures are here on the left, where there's very a male-biased power. And then the countries here on the right are much more, this would be, much more gender equal countries. And what you can see in terms of the sex difference in age preferences is they decrease as gender equality increases. So this is the sex difference here. These are females up here who would prefer partners that are about 2.5 years older than average, and these are the males here who would prefer partners that are about 2.5 years younger than them. And what you can see as you get more gender equality, males are preferring partners that are closer to their own age, and females are too. So yes, there is an impact, you're absolutely right. But we still see a sex difference.
Yes.
So will it disappear completely? Who knows? But this is the idea that our psychological mechanisms may be tuned to the local social environment. As the social environment becomes more equal, mate preferences seem to be becoming more equal as well. You're absolutely right.
Thank you.
Yeah.
Thank you for your great talk. My questions come from ignorance. I'm not a biologist, I'm not a psychologist, I'm a linguist. I'm here by chance, so to say. But I have some questions, mainly basic questions. One is, which is the, let's say, ontological nature of this gene regulatory networks? It is something biological. This is the program built in the gene.
Yeah. So what this is.
The other one is, which is the difference between this approach in which you talk about tuning to certain conditions and epigenetics?
Epigenetics, is that what you said? Yeah. So the networks, that's a great question. Where do they come from? So we can think of our DNA as a linear string of 3 billion symbols, A, C, T, and G. And this linear sequence is divided into what we call genes. So each gene specifies the structure of a protein. And those proteins, some of those proteins, not all of them, but some of them are what we call transcription factors. And those proteins, those genes that code for transcription factors, the transcription factors have a special structure called the DNA binding domain, and it's like a key. And so it will only bind, so the this gene produces a transcription factor, which is a protein. The protein has a special section of it that has a kind of a key-like structure that will only bind to certain areas on the DNA. And when it binds to those areas, it causes that gene to either produce protein or not produce protein. It kind of turns the gene on or off. And different transcription factors have different keys that fit different parts of the DNA. So the gene regulatory network is the set of these transcription factors that have specific binding domains that connect to different parts of the genome, which then connect to different parts of the genome, which can connect to different parts of the genome. So it's that pattern of interconnections determined by this kind of lock and key system that determine the networks. Does that make sense? Yeah. So that's where the gene regulatory networks come from, is this pattern of binding here. Now, in terms of epigenetics, epigenetics is a rather generic, general term, and so many biologists say, this is epigenetics right here. This is the genome these are proteins regulating itself. Another aspect of the epigenome would be that, and this is I didn't get into, but DNA, about 1.5% of it actually codes for protein. Some of those are transcription factors. But a lot of DNA gets transcribed into RNA, and you have to know what RNA is. But basically, the chemical process goes from DNA to RNA to proteins. But you also can have DNA to RNA and not going to proteins. This is called non-coding DNA. And that RNA can also bind and regulate the expression of genes. So that makes, we have another element that I didn't talk about here, the RNA elements, the non-coding RNA elements that also regulate the genome. And then there are some other things like methylation that can bind to some of those ATCs and Gs, and again, turn on or turn off genes. So yes, epigenetics is definitely part of this. It kind of depends on your definition of epigenetics, but right here. So we definitely have mechanisms by which things can influence the expression of genes, elements. We can have chemicals in the environment, as we saw with these guys. Just chemicals that are out in the environment can alter the behaviors of these gene regulatory networks. But there can also be products, other cellular products, proteins, RNA, other ligands produced by the body that can alter the behaviors of these gene regulatory networks, absolutely. That's critical to the whole concept. Did that answer the question? Sort of? We can talk more if you'd like later.
Now, it's kind of a continuation of this, what you just said. No? But for me now, after your explanation, made me think, even got a bit confused, some concepts you showed. Like, for example, what is the difference between genetic switches and epigenetic switches?
Yeah, I think that's a good question. So, and again, it really depends. Unfortunately, epigenetics is a term that's not very precise, and it really kind of means so genetic, everybody would agree transcription factors are produced by genes, they produce proteins, they regulate, so they may exclude transcription factors because those are genes regulating genes. But what about the RNA that is not, in some definitions of a gene, it's not producing any protein, it's getting transcribed, but it can also regulate DNA. And what about methyl groups that get imprinted, that attach to certain parts of the genome, that can also alter expression of genes? And then our genome is wrapped around, these are called nucleosomes, we're wrapped around these proteins, so there's a packing structure. And those protein packing structures are called histones. And to actually express DNA, you have to kind of open up the DNA, open up this package, and so you can get histone modifications that are modifications of the packing proteins that can also alter. So your question is actually a good one. How do we distinguish genetic switches from epigenetic switches? It really depends on which lab you're working in and how they want to define epigenetic. It's really kind of an unfortunately vague and ambiguous term. I saw something on Twitter recently that had I think it was a 4x4 grid of various epigenetic definitions. So there's 16 different definitions, and depending on which tradition you were in a different cell. So I kind of don't like the term epigenetic because it's just not very precisely defined. That's about as good as I can do for that, to answer that question.
Você traduz? Eu vou falar em português, você traduz. Eu queria saber, na opinião dele, o quanto ele acha que nós, psicólogos evolucionistas, estamos conversando bem e fazendo pontes com as pessoas que fazem epigenética, porque eu não acho, eu acho que é muito gene centrado ainda essa ponte que a gente tenta fazer.
She wants to know what you think about the relations we as researchers in development, no, in evolutionary psychology.
I will try. So,
okay.
We, as evolutionary psychologists, in your view, in your opinion, how do you think if you are doing a great job in in building bridges with, I don't know, epigenetic colleagues to talk, I don't know, because I think we are very much gene-centered. Even though we know that epigenetics are in the picture, but in my view, I think we must building much more bridges, talks, and understand much more to, I don't know, talk about this kind of thing, because I don't know, I consider myself much ignorant in this matter to know, I don't know. There are a lot of definitions in epigenetics, but I consider myself ignorant. I have to study much more. Do you think that we can build a strong build with this area, or we are in our infancy to build this?
So what we see right now is we've and I'm not sure if I fully understand your question, so feel free to...
Sorry about that.
That's okay, no. Feel free to jump in and steer me on the right course if I start heading in the wrong direction here. But, you know, in the last couple of decades, we sequenced the entire human genome, and we've now developed technologies for sequencing the entire genome for a few $100s. So we now have tens of thousands, maybe even hundreds of thousands of complete genomes. And that has given us, we basically now know what genetic differences look like between people. And that really hasn't taught us very much about what it means to be human. And what you see if you just dip into the literature is that there's a massive amount of research to try and understand these gene regulatory networks, to understand gene expression. And they're developing what are high throughput techniques to look at cell by cell by cell, which genes are being expressed in which cells and when. And it's a vast amount of research, but it's extraordinarily complicated. We're very much in the very, very early stages of putting this together. So even with, you know, these guys, sea urchins, we can only do the first few hours, and even then we're not 100% sure what's going on. And this is a system where we can actually, it's ethical to manipulate and knock out genes. So yes, we're in very, very early days of understanding this. So even though I think there's good reason to believe this will be the foundation, and maybe for very limited problems, it might help us. I think the real value for me of evolutionary psychology is this picture, which is if you looked at psychology prior to evolutionary psychology, it really wasn't asking what I would consider to be fundamental. How did humans survive and reproduce? What kinds of mechanisms do we need to solve all these problems? So there was all kinds of things on cognitive biases and sort of various ill-formed questions about social relationships, but it really wasn't getting at fundamental problems of survival and reproduction in the kinds of environments in which we evolved, which are often very similar to the problems that other animals were facing. And so for me, the value is really thinking about this and how this might explain universal psychological structures as we saw in like these examples, like this. I love this plant research and infants just somehow innately knowing that plants might be dangerous. So I think for me the value of evolutionary psychology is kind of turning the ship around to begin asking the right questions. It doesn't necessarily give us the answers, but it's helping us ask the right questions, I think.
Thank you so much.
Yeah.
I have one more question. I'm sorry to keep bothering you.
Go ahead. No, that's fun for me.
So now, one thing that I didn't understand and is still in my mind that you said at the beginning of your presentation is about the like 95% of DNA is noise. What do you mean by that?
Great question.
It's junk.
Yeah, great question. So, there's a technical term called conserved. And what conserved is, we know that mutations happen randomly. And we have a mathematical model called the neutral model, which says if mutations are happening randomly and there's no positive selection, there's no negative selection, then the statistical patterns of genetic variation should match what's called the neutral model. So it's basically a model of noise. And what you can do is you can look at, we now have all the genetic data from large numbers of humans. You can look piece by piece across the genome, and you can look at the patterns of genetic variation, and you can say, are those patterns consistent with what's called genetic drift, or just random mutations that are happening that are not being positively selected or not being negatively selected. They're just happening, and the kind of random processes that, and if that's true, that means that those areas of the genome really are probably not having any influence on the phenotype because they're not influencing survival and reproduction, therefore there's no selection, therefore the patterns look totally random. And so that's what we would call not conserved. And when we look at the genome section by section by section, it looks like about 85% of it, or maybe 90% of it, is evolving neutrally under drift, that mutations are accumulating randomly, and they're not undergoing positive selection, they're not undergoing negative selection, which means they probably just don't have any impact on the phenotype whatsoever. And only about 10% of the genome looks to be conserved, which means that if a mutation happens in that 10%, it usually will be eliminated by natural selection. So what we see is 10% of the genome is surprisingly missing genetic noise. There's no variation. You and I would be identical. So if there's a gene where you and I are identical across the entire, all the, let's say, 500 amino acids in the gene, it's probably because that if any one of those changed, we might not even get through development, or we would not survive and reproduce. That's called conserved. So about 10% of the genome looks like those are areas where when mutations happen, they're quickly eliminated by natural selection, which means they have an important impact on the phenotype. So when we look across the entire genome, it looks like, and there's still a debate about this, so I don't want to put an asterisk by this, but it looks like about 10% of the DNA is critical, and whenever mutations happen in that part of the DNA, they're quickly eliminated by selection. But the other roughly 90% of the DNA, when mutations happen, they just happen and they stick around, and they don't have much impact on the phenotype whatsoever. Or, as this diagram showed, they may have an impact, but it's very minor. So most of these genetic variants are rare, which means these mutations have happened. They haven't been so severe that they've prevented the fetus from developing or the individuals from surviving and reproducing. But they're, so they just they're either what we call neutral or very mildly deleterious. Did that answer your question? Sort of? Sort of? Okay. So basically the conserved parts are the important parts that really influence the phenotype, and the non-conserved parts really don't influence the phenotype, and it really don't have a big impact on survival and reproduction.
So thank you very much for the discussion, and thank you, Ed, for accepting this invitation, for giving
my
us
pleasure.
this wonderful talk and discussing with us in the meantime. And we
thank you.
brought you a small present so that you can do your own caipirinha at home,
Oh,
which
yes.
always helps, right? We need to increase the well-being of our speakers. So thank you again. And now we will have a small coffee break. Try to be relatively quick, I think 10 minutes, 15 minutes in maximum, and then we will go for the last session of today. Mostly about sexuality. So we have some 15 minutes for the first presentation. Irene Del Val.
Okay. Thank you, Thank you, Yarka. Thank you very much to everybody. Good afternoon. Boa tarde. Buenas tardes. Thank you very much for being here in this traveling experience, time traveling experience. It's a great pleasure to be here today because I know the difficulties for organizing this meeting. So finally it's happening. So I'm very glad that it's occurring. And I am the first one in this session about behavior, sexual behavior, the second thematic session of the Congress. My name is Irene Del Val. I'm from Spain, but I have been in Brazil for more than 10 years now. I did my master's and my PhD here, and now I am a postdoctoral fellow in Yarka's lab. So I'm going to show you a small piece of our work there in Yarka's lab of my postdoctoral project. So today I'm going to present to you a case of homosexual courtship in wild Capuchin monkeys. So I like to start talking a little bit about homosexual behavior in animals. It's a topic of great interest in the biological sciences, of course, but it has been, for a very long time, it has been obscured or not shown because of moral or religious issues. The first records of homosexual behavior in animals was not reported because people thought that they were possessed by devil or things like that. So it wasn't a topic of interest at the beginning, but now of course it is. But however, for many reasons, it's often considered as well as a Darwinian paradox, since selection might act against traits that are not enhancing reproduction. But we don't know if that's really true, because this trait, not because, but this trait, the fact is that it is ubiquitous, not this trait, this behavior is ubiquitous in the animal kingdom. We can find it in more than 1,500 species now, going from insects to birds, or of course mammals, reptiles, etc. So it's all over the animal kingdom. We can see it everywhere. So we have to explain it and think about it. I always like to explain that when we talk about homosexual behavior in animals, we are not talking about homosexuality, because this word, it involves another concepts, different concepts, and more permanent tendencies that we cannot ask to animals, we cannot post in animals. So we prefer to use homosexual behavior, or the expression we usually use when we refer to homosexual behavior in animals is same-sex sexual behavior, which is the term that I'm going to use through this presentation. In primates, same-sex sexual behavior has been documented in most species. Today, in more than 51 species have been reported, including prosimians, monkeys, and apes. At the beginning, prosimians were not reported to have to perform same-sex sexual behavior, but today there are lots of records of prosimians performing same-sex sexual behavior. So it's not there was a phylogenetical explanation about that it's not posted anymore. So, but another important fact is there is a huge intra and interspecific variation in the frequencies and in the forms this behavior takes place in different species. So we can find different functional explanations for this behavior according to the species or to the age of the individuals. So for instance, which is the button here? The typical form of same-sex sexual behavior in Japanese macaques is ventroventral mounts among adult females. While in bonobos, bonobos, they are famous for their rich sexual behavior, and you can see it's frequent in every age class and more frequent ventroventral mounts. And there are many other different same-sex sexual interaction according to the species and many different explanations. in Capuchin monkeys, which is the species that we study here. If you have seen the symposium this morning of Patricia, Briseida, and Nicola, I study the same monkeys they study. So in Capuchin monkeys, same-sex sexual behavior is typical from juvenile play. It's, they perform a lot of ventroventral mounts during play, common among males, and it's often explained by the need of practice hypothesis, which suppose that young individuals need to practice for further heterosexual performance. However, same-sex courtship, which is not just a mount, a sequence of mounts, has never been documented in this species. So today, I'm going to show to you, this is a small piece of my postdoctoral research, the first report of same-sex sexual behavior in Capuchin monkeys that includes a complete courtship display. This is an outstanding fact because this behavior has never been reported in Capuchin males, and also because one of those individuals is very, very young. He's 18 months old, and he is still nursed by his mother. So this gives more relevance to this record. But before showing, I'm going to show to you a small part of this record, which consists in a 15-minute uninterrupted sequence of sex and love. But I would like to introduce to you the courtship display, because it's very interesting, the courtship display in Capuchin monkeys. They are known, they have been named as erotic artists because they perform a really extravagant and I don't know, dramatic sequence of behaviors. So, and they have been already really well studied. I like to point out, for instance, that this draw from the first treat of natural history, the first long treatment of natural history, represents the typical specimen of the Capuchin monkeys with a perceptive female. You know, this is the typical face of the perceptive female calling attention of the male that Briseida called, that she said that they were sticky. So these are some representative behaviors of this species courtship behavior. The female, it has four faces, and the female begins solicitation male's attention, and then the male goes into the courtship and responds, but it takes a while for doing that. Then eventually it's mating, and sometimes, not always, it occurs a post-copulatory display. These behaviors have already been published and described, and they are in literature, but only for captive Capuchin monkeys from the species Sapayus apella. So here I'm going to show to you here, I don't know if it's going to work, a brief piece of my record of this homosexual courtship. And you're going to see Pimenta and Dumbo. I don't know if it works. But you cannot listen. But these are Pimenta and Dumbo performing most of the behaviors we have record for. Well, this is a spoiler from the regular typical heterosexual courts. So what we did is taking this setogram from the literature and apply to our observation. Two independent trained observers analyze the video, applying the setogram, and we transcribe it with a specific software. It's used for behavioral analysis of the observer. So here, you can see there is one that is a sub-adult male, and there is the other one, Pimenta, which is 18 months old. Look, he's making the typical female face, like opening his mouth. Well, this is a very short sample, but this is a very interesting record. So what we found
is that the two monkeys performed 16 out of the 20 typical behaviors of the heterosexual courtship, and that it was similar to the second phase of the courtship, the mutual courtship phase. There were 4 behaviors that are typical from the heterosexual courtship that they did not perform, and there were also 4 novel behaviors that we will further study, since we need to know if they are typical from young individuals or they are typical from this species, since this is a different species from the one that is reported in literature. So there are things to understand. And some topics from discussion about this fact is why these two young males are performing this homosexual courtship. It's very interesting because they are not both, but the youngest one is outside of the hormonal triggers that give the starting point for sexual behavior. So it's very interesting, and we have this discussion. It's opening questions that we need further research. Is it a rehearsal or it has another function? What we know, it's a powerful way of communicate social messages. So I suppose that this is not a practice. No. Sorry. We are supposing that this is not practice, because this is the first time this behavior is recorded and reported. And I have been on the field for years, and I have already analyzed hours and hours of video records, and this is the only record of courtship behavior. So if it's a practice behavior, why it's not everywhere and every day among young individuals? If they need to practice it, it must be more frequent, right? So it has some contributions. This record has some contributions to do. That is, it's the first report concerning wild robust capuchins, and it's the first description of courtship among 2 males, the first courtship among immature individuals. And now I would like to make a little bit of propaganda of my own project in order that you know what we are doing now here. And so my postdoctoral project is about the development of sexual behavior in wild robust capuchin monkeys. And this is the result of the collaboration on these 2 wonderful women, which are Patricia Izar and Yaroslava Varela-Valentova, and the collaboration of these 2 labs, my former lab, ladies, in which we used, and they actually continue to try to understand developmental issues and social interaction of capuchin monkeys and other animal species. They study also domestic dogs, and the contribution of Yarka with her lab of evolution, behavior, and sexuality. So by these bridges, this collaboration, we are creating a new area of research, which is primate sexuality, primate sexual behavior in the University of São Paulo. So our main goals, we want to understand when does sexual behavior begin. There are many possibilities. It can, in capuchin monkeys, of course, I'm talking about capuchin monkeys, it can occur just after birth or only in adolescence. We want to know also which are the first sexual behavior, there are many, and which are the first preferred sexual partners, and if this changes along development, because it's not, this is, of course, you are acquiring proficiency and everything changes. Which are also the first sexual context, and also if there are any intersexual difference. We expect that there is not going to be the same thing for females than for males, because sexual conflict and many factors. And also, since, as I'm going to explain next, we are studying 2 different species, we want to know if there are ecological differences. So we do that by studying these 2 species, Sapajus libidinosus and Sapajus xanthosternus. One of them, xanthosternus, is critically endangered species. They inhabit 2 very different habitats in Brazil. They are both endemic from Brazil. One of them inhabits, as Patricia said this morning, a semi-arid in the north of Brazil, in this region here. And the other one, Capuchins sternus, lives in the Atlantic forest. It's a very different area. It's close to Amazon. Okay, I'm finishing. I'm sorry, I'm almost over time. So what we do is that we record every week. We go to the field and we film the individuals. We have focal animal samplings videos from the first week of life of these individuals, and we have storage them in a database which is available for new students. So we do that. We have, for this project, we have selected 16 individuals, but we have many more, and we are going to study the first 3 years of life. That's not important. And then this is a very high demanding task because we have to collect the data. I've been on the field for almost 2 years collecting data. Then we have to classify data, transcribe the data, and of course analyze the data. So we need people, and we invite people to write to me in order to acquire, to give response to some of these questions. We want to, in resume, I would like to say that we want to know what is sexual behavior and improve its definition, because it is not limited to mounts and genital contact. There are a lot of details that we want to understand. We want to understand when does sexuality begin, and we can study this in a primate species far from taboos and things like that occur with children, so we can understand which are the first sexual partners without thinking about difficult stuff like pedophilia. We want to understand what other functions sex has in other species, and I would also like to highlight that same-sex sexual behavior may enhance fitness because it has a very important functions in social communication. So please never forget, we are all primates, and then comparative approach is very important for understanding our own behavior. And also, I need to thanks to the founders, because without FAPES, PICAPES, at least it's not possible to perform this high demanding task and research. And thank you very much to you all here for being here, for listening to me, and to the organizers for letting me present my work. Thank you very much.
Thank you, Irene. Very nice presentation. Are there questions, comments, suggestions? Thank you. That was a really nice presentation. And I want to take you back to your questions because, you know, you suggest that the last slide that sexual behavior was about communication and love.
Well, maybe that's, yeah. Not only for that, but it's
but
used,
can you expand on that idea? I love
used
it.
for that. Well, that's maybe I need to prove that. Maybe it's an insight I have because I see it so frequently in nature. So that I see those individuals so exciting that I need to analyze that. I see that like an expression of love, their excitement, and of course it has many social functions and communication, since it communicates behavioral states. There are the dominance hypotheses when someone mounts the other one. But I really do believe that when they are performing sexual behavior among individuals like these ones here, they know each other well, they are connected, and they are through a sexual interaction which is about taking care of both of them. But I don't have a clear explanation for that. Thank you.
Other question? Last question.
First, congratulations to your work. It's very nice. Yeah, I would like to ask you if you see, I can't to speak now, a female-female interaction. And one question, female-female interaction, and second, why did you think it's so rare? Why did you didn't observe more? No, in the beginning you showed you found this homosexual behavior in 51 species, no? And you only present this case. Now, why is it so rare?
Yeah, thank you very much. It's a good question about females. I've never recorded female-female interaction, and I have been in the field for, as I said, for a year and a half, and never record and neither see female-female interaction, young female-female interaction, because my focus has always been young individuals. So I have records and I have been watching juvenile males and females. Never record female-female interaction. I don't know why. I have seen adult females performing some sort of not sexual, but that's the question for improving the definition. Some kind of behaviors that can be part of courtship behaviors, like touching each other breasts and things like that, that are considered when they are within the sexual interaction, within courtship, they are considered like sexual. But it's not. I have never seen robbing, for instance, as we see in bonobos, for instance. But about the other part, it's so rare. Which is rare. It's the courtship behavior. Courtship is a very paternal display, as it has never been recorded. Same-sexual mounts are all over. Males are all the time mounting each other. The Capuchin juveniles play. It consists in rough and tumble play, and then it ends in a mount. But it's very different because there are two individuals playing, and then a mount, and then another one gets in, and it's very quick, and it's a very different stuff. This courtship behavior, it's similar to the courtship behavior of adult male and female, and it has never been recorded for two individuals pertaining to the same sex, neither adults or immature individuals. So the particularity is the courtship behavior, which is different and which is rare, because same-sex interactions like looking at each other's genitalia or mounting, it's very common. Did I answer? Okay. Something else?
Okay. So thank you very much again for a nice presentation. And we will go for the next one. Who is next?
Lucas.
Lucas. Where is Lucas? Lucas is there. So our next can you find him?
Is it here? ¿Es aquí, Lucas?
So next short communication has a very interesting title, The Sociosexuality Explained. So I really want to see that. Anyway, and the presenter is Lucas Schirmer, who is student from the Catholic University in Rio de Janeiro. So it's your turn.
Hi. Thank you everyone for your attention here. First of all, I would like to thank the organization for the opportunity to talk to you today. My name is Lucas. I'm a graduate student in psychology at PUC-Rio. And I'll be presenting to you today some data that we collected on our study on social sexuality and Tinder behavior. So we call this study the Social Sexuality Explained, what the gender and targeted relationships on Tinder reveal about sexual commitment. First, I'll be talking to you a little bit about our background theory. I'll be talking a little bit about parental investment theory. It is a theory that basically explains that most animals, species that we study, when offspring is newborns, they need to be taken care of, primarily by the father and mother of this newborn child or any other animal. So we might think that both parents need to invest resources in the taking care of this child, but gender plays a big role in what kind of investments are going to be displayed. So most mammalian species, and humans are no different, we might think that women need to expend more direct resources in taking care of newborns and their offspring, because they have to take care of the offspring on their belly for a really long period of time. They have to breastfeed. They have to spend a lot of resources. Okay. And they, on the other side, males, they need to spend resources as well in order to their offspring to thrive. But these resources are mostly indirect resources. They're not direct. So we might think someone might invest in getting relationships in a group setting, or in providing food for their offspring later on. But we really need to think of these two different aspects of direct and indirect resources that are explained. And also, I'd like to talk a little bit about social sexuality. We can think of social sexuality as a psychological construct in which we think of individual difference in the willingness to engage in sexual relationships, sexual behavior, outside the boundaries of a committed relationship. Individuals vary on this degree, as I will explain more later. But for now, that's what you need to know. And online dating apps. The most worldwide used online dating app is Tinder. That's why we chose it for this research. And people use these dating apps for different purposes, different reasons. But their behavior reflects what our theory engaged. So the first objective of this study was to verify gender difference in what is sought for on Tinder. And the second objective was to test the interaction between gender, the kind of relationships, and the levels of social sexuality. And for this study, we collected data from an online survey that contained the social sexuality orientation inventory revised scale, the Brazilian version, which is the most used scale to assess social sexuality. This is a 9-item scale with a 3-factor structure underlying it. So we think about social sexual behavior, attitudes, and desire. And also we have a lot of questions about how people use Tinder. People might use Tinder for search for casual relationships, for committed relationships, for friendships. They might use it just because they are bored. It doesn't matter. But we asked people why they were using and how they were using this app. Again, here you can see a normal distribution of social sexuality, where in one side of the distribution, people are more restricted to engage in sexual behavior outside the boundaries of committed relationships. And on the other side, we can see people who are less restricted. participated in our study 2010 adults. 53.3% of them were women, with an average age of 23.2 years old. In this research, we only utilize people who consider themselves either male or female and who were heterosexual. So the results. Just a minute, please. Okay, here. So, okay, so technical problems here. Okay, now we're back. Much better. So, first of all, in order to fulfill our first objective, which was to, again, verify gender difference in what was sought on Tinder, we asked people if they were using Tinder to search for casual relationships, committed relationships, or friendship. And we realized the chi-square test, and as you can see here on first table, it was deemed significant for 95% confidence. And on the second table, you can see the effect size. It was 0.17, and it's considered a small to moderate effect size, but as I said earlier, is statistically significant. So it's a positive result in this direction. And we can see that when searching for casual relationships, men are much more willing than women. But on the other side, when searching for committed relationships or friendships, then women are more represented than men. We see this different pattern here. Now I'm going to show you three graphs that show the different average level of social sexuality for men and women on these three kind of relationships they were searching. So casual relationships, committed relationships, or friendship. I will not talk too much about friendship because due to lack of data, not many people said they were using Tinder to search for new friendships. So I will focus my analysis here on casual relationships and committed. Here is the SOI behavior factor. And as you can see, the mean score for males on casual relationships was significantly higher than women. But there was no statistical difference on the committed side of the spectrum. Next, we can see the attitudes aspect. And here we can see the same pattern. So when looking for casual relationships, men score higher than women, but not when searching for committed relationships. And last but not least, when looking for the desire aspect, we can see that, again, men scored significantly higher than women when they were searching for casual relationships on Tinder, but not when they were searching for committed relationships. And what we can conclude for this data that I presented to you. So, first, the parental investment role. We might think that gender difference regarding the differential parental investment styles, as I was saying earlier, they impact the motives for people who use these apps. And the second conclusion is that we might understand social sexuality better by looking at both gender of the person, but as well as what kind of relationships they are trying to search. And that's all I got for you today. Now, thank you everyone for your attention, and if you have any questions, I will be glad to answer them.
Any questions? Suggestions? Timon?
Hello, Luca. Thank you very much for your presentation. It was just a wonder about your recruiting. You sent a questionnaire, and I wonder if you had a special way to recruit people, if you targeted special medias to find people who were using Tinder in particular.
Thank you for your question. So this questionnaire, we used an internet collection method of data collection. We did not use specific search methods for searching for these people. This might explain why we didn't have too many people who said they were homosexual, for example, that might use other dating apps, not Tinder necessarily. But it's a good point. Thank you.
Lucas, your participants could choose the two options they were looking for, casual relationships and committed ones, and you have analyzed this?
Yeah, they could choose if they were seeking for casual relationships, committed relationships, or if they were looking for friendships, to make new friends. But as I said earlier, not too many people said they were searching for new friends. So that's why I did not speak too much of this aspect. They could choose both, no. Not in this study.
Thank you. Lucas, I was wondering if two questions. If do you intend, Jean and you, to amplify your sample, like focusing in much more like homosexual, and actually you are talking about gender, but I think you are just considering male and female. So maybe there is a huge discussion in our area about sex and gender, and so I think we need to amplify and search for to recruit much better our samples. Afterwards, I was wondering, and I'm very curious about, did you control for age in your study?
Okay, first about sampling. Yes, that's a fine question. It's very important for we to search for people who are not these normatives like men and women, heterosexual. Sometimes it's difficult to get to these people. We could make new studies in this direction. Now we're not doing it right now, but maybe in the future. And it might produce interesting results. And about controlling for age, in this graph that I present to you, I was not controlling for age, but it might bring some new insights, because certainly age affects how people choose their mates and what they value more, which traits they value or devalue.
And there are a lot of apps, so there is Bumble, there are a lot of different filters, and maybe you can deep your demographics, do you know? Tinder is, I don't know, I think it's worse than Bumble.
It's worse than what?
Just one idea. Just one.
Yeah, for sure.
All these experiences. Thank you very much, and we will go for the next. Oh, is it me? Thank you. Muito interessante. Obrigada. Você é o último, Bruno?
Depois a gente faz. So we'll continue. My name is Jaroslava, or everybody is calling me here Jarka, so that was me. But my real name is Jaroslava, from the Czech Republic, but I work here at the Institute of Psychology, the Department of Experimental Psychology. And I'm actually really happy to have all of you over here. So thank you again for your presence. And I will be talking just a little bit about appearance modifications, which is now recently one of my favorite topics. And I will try to be fast because there is lots of things that I might talk and I would spend hours on this topic. So, okay. Not working. Right. Yeah, not even the organization can do that. Wow. Hmm? Não consigo nem tirar. Funciona isso? Não funciona nada. Ah, travou aí. Ah, que travou.
Christian.
Ah, calma. Ah, não sei o que aconteceu.
Mas esse negócio não está funcionando. De que modo a apresentação
Não funciona nada, eu só quero fechar. Será que agora vai? Agora vai. Muito bom. Yeah, the technical part. So let's start again. So appearance modifications. So probably everybody modified their appearance somehow today already, probably many times since you woke up. So it is something that we normally do in everyday lives. Why people change their appearance, that's actually the topic that I want to discuss with you today. It is actually very interesting because there is a high frequency of people who are not really satisfied with their appearance, and that's why they might be trying to change how they look. But there might be also other functions. And I will go through them actually quickly. It's interesting that most of the research, even mine own actually, when I was studying facial appearance and attractiveness, we were usually focusing on morphology, and we were actually eliminating any kind of sociocultural appearance modifications. But actually we have never been really a naked ape, right? So it is interesting to see how people are working with their own appearance and trying to change their appearance, and why. And there are many functions, and I will mention just I think that's because it's the last session, right? So the batteries are already over. But anyway, so there are several modifications, several functions of modification, such as attracting of potential partners. That's usually the first one that pops up in the mind of people who are thinking about why people are actually trying to look better. So they would attract someone who is actually more attractive or of higher mate value. And that definitely happens in many different ways in each culture, probably differently. But there is also the side of intrasexual competition, which used to be overlooked in this kind of research or reasoning. We don't want to only attract attention of potential partners, but we also want to beat our rivals so that we would look better than those who might be actually attracting attention of the same potential partners. There might be many explanations for this, such as good genes. It might be also Fisherian selection, so it can be like really random fashion. But it can be also sensory exploitation. We might like things that actually are not, that didn't really evolve to attract partners. There might be also supernormal stimuli that are actually attracting attention because they are simply more than natural. Other theories would probably say that the social mimicking is actually very important. It doesn't really matter if you are in Brazil or in Korea or in other country, but mimicking the models, the successful individuals might have some social and evolutionary value. So changing their appearance, as this nice example, one of those two guys is the original Korean pop star. The other one spent millions of pounds to look like the pop star. And yeah, well, that's what people sometimes do, just to look differently and better, at least in their eyes. But of course, there are other reasons or other functions why people are changing their appearance, such as social affiliation or group affiliation. By changing our appearance, we can easily show to which group we belong and to which group we don't belong. So on this example, like around here, which I, of course, can't do, no. Anyway, we would see probably easily who is the fan of Brazilian football and who is the fan of the Czech football, just looking at the crowd, right? So people are actually changing, or we would know exactly who is the punker and who is the policeman, and also what are their roles in their society. So it's actually, right, that people are not only showing their affiliation, but also the roles in their lives and in the society. By changing appearance, people are also advertising their status, so they can show if they are of higher or lower social status. Again, all this depends on the local traditions and local environments and conditions. But the mechanism behind those, right, changes is basically the same. Of course, we are also changing appearance only during specific time periods. For example, during weddings, right? People dress during a wedding ritual totally differently, and they would never use the same dress or makeup or shoes as during this day, in any other day during their lives, or when they are reaching maturity, when they are actually trying to get out of the world of children or infants or subadults, and they are actually entering the world of adult people. Or when we are gaining a degree, we also, or in many cultures, not everywhere, probably we are doing some kind of ritual, such as in Prague, where we were even promising something in Latin. Nobody knows what we were promising. Or this small guy who is actually waiting for circumcision in Turkey also is dressing very specifically and only will be dressed like that during this ritual. Of course, there are other functions, right, such as protection and camouflage. And mostly when zoologists or researchers who study non-human animals talk about appearance modifications, mostly they are actually talking about this kind of functions. Right, so, but in people, these changes, such as clothes or using shoes, can also have many practical functions, such as thermoregulation or protection against parasites or against sun, for example. appearance modifications are also important as a part of self-grooming. So people are actually touching themselves and other individuals, sometimes with needles, knives, and this kind of quite threatening objects. So actually you need to trust the other person. So it is strengthening the social bond with the other person who is, for example, doing the tattoo or other modification. So, right, one of the functions can be social bonding and maintenance of social bonds. Of course, sometimes people go over the healthy limits and they change their appearance too much. They spend lots of money, energy, and sometimes even their whole lives changing their appearance so that they would appear like completely differently, sometimes even as a basically different species. Virtual changes of appearance are still rarely studied, but of course, nowadays it's probably the most practiced appearance change, so we still have lots of things to learn actually with all these additions or picture additions, what we do, what people do, why they do that, if it is appreciated by others or not. And sometimes some studies even show that actually it is not as much appreciated by others than we think. I based myself basically on a review article about appearance enhancement by Ednoski and Davis, and they were talking about many different kinds of appearance modifications by tanning, by makeup, by hairstyling, by tattoos, even exercises, piercings, depilation, right? Many things that actually come to our mind when we are thinking about appearance modification. However, we thought that there was something missing in their model or in their review of the literature. And they were actually talking about visual appearance enhancement. And we thought that this is actually only part of the whole picture, and we suggested that visual appearance enhancement is actually part of self-presentation enhancement in general. Sometimes we are not only enhancing or improving our appearance when we are doing this appearance modification. We can, by applying cosmetics, for example, we are also improving our olfactory presentation, right? So, or tactile presentation, right? When we are, for example, depilating legs or other parts of the body, actually it feels differently when we touch the person. It's not even visible sometimes. So we suggested a more broader model of self-presentation enhancement that might be actually interesting to study in this perspective of evolutionary and social functions. When we think about it in a broader sense, then we can see also other species who are actually doing lots of appearance modifications, such as this beautiful orchid bee who is collecting lots of fragrances, and in the mating season it's actually applying these fragrances on own body to attract other individuals, which is a beautiful modification, but it's not really appearance modification. It's right, it's olfactory self-presentation modification. And to make it even more complete, this model or this picture of appearance modifications, we suggest that actually sometimes individuals do not want to improve their appearance. Sometimes they can change their appearance to look worse. Why would they do that? Why would people want to look worse? But sometimes actually they do. They want to scare off other people, or they want to blend in, right? They don't want to always appear, we don't want to always appear beautiful and attractive, because that might be too much. We don't want to always attract all the attention of all the world. So sometimes people just want to blend in with the crowd. And sometimes parts of many rituals is actually scaring. Well, sometimes scaring the children, right, or other people, exactly not to attack, for example, to prevent some kind of conflict or war. And yeah, That's probably it, just to show you that not always we are actually enhancing our appearance. Sometimes we actually want to look really, really bad. Thank you very much. If you want to ask something, you can. I think we have space like for one question. And I go.
I was just wondering if you would give any examples of what hypotheses you might have for when you would dress to impress versus to make people feel averse towards you. Like, what would be some examples?
What would be examples of worsening?
Yeah. Maybe one day you do want to look nice, and other days you don't want to look nice. What type of, if it's a frequent enough thing, you might be able to predict when you would go for one versus the other.
That's actually a very good question, right? This is a theoretical model which we were trying actually to show the complete picture, right, of appearance modifications, but we don't have any data on worsening of own appearance. There is no research about it. What we know, for example, that the detection of ugliness or non-attractiveness is much quicker and even more universal than detection of attractiveness, right? So this is actually interesting, but we don't really have data on why, when, under which conditions people are changing their appearance for worse.
I was thinking something like, to avoid harassment from the opposite sex.
Yes. It can be avoidance of harassment, which actually it appears also frequently in models, right, or in people who are really highly attractive, that they are actually trying to really blend in. Otherwise, they would be pursued all the time by potential sexual partners, unwanted sexual partners. But it can be also like revolt against the society, right? Against the patterns of society. I mean, everybody is telling me that I should look beautiful, so I won't, right? Which was exactly the movement of punk, for example, but many others, like in rock bands, there were actually many, specifically female or girl rock bands, actually worsening their appearance, showing that we would go against the crowd, right? So it can also have this aspect of going against.
I'm wondering if one category that might have been missing, I'm not sure, is in rituals, you modify your appearance to play a particular symbolic role in a ritual. So would that didn't seem to be in your scheme, but maybe it was the symbolic use of appearance modification in playing a particular role in a particular kind of ritual, for example.
Yeah, that's quite possible, actually. We should, to complete it, right, we should include a timeline, because sometimes some tendencies are probably more stable during lifetime, but some are really punctual. And so that would be really interesting, right? What people, but actually we did one study specifically about makeup usage in women, and we were asking them in which situation they use makeup more than normally. So when you go out with male friends, with female friends, when you go for a first date with someone, when you go to an interview with the boss, and things like that. But actually, we didn't find much individual differences in makeup usage. What we actually found was that some women were using more makeup than others, and it didn't really matter which situations. But of course, this is makeup usage. This can be different than, like, I don't know, using some specific costumes which are really very, very specific for some situation, for some ritual. Yeah, that would be actually interesting to see if there are some patterns, right, for some like specific social or rituals which have specific social roles.
Yeah, like the hunter-gatherers I work with have these special rituals and special costumes that go with each one, and then various take on those roles and dress up in the particular creatures that play a role in the ritual and things like that.
Yeah. This is actually very interesting because there is lots of rituals where the people are actually pretending they are other species, for example, right? Yes. Yeah. Okay, I think that's it. Thank you for the discussion, and we will be discussing more during the cocktail. And we have the last presentation. Who is Bruno, and he's actually a PhD student from my lab, from our department at experimental psychology here at University of São Paulo. And nothing. Sumiu total.
Hello, good afternoon. So my name is Bruno. It's a huge pleasure to be here presenting my PhD project for you. This is actually my first presentation at a Congress, and it's especially amazing because it's an international one. So yes, it's a huge pleasure to be here. As Yarka was saying, I am her newest PhD student, and my project is still at a very preliminary level, but it will be a pleasure to share it with you. So the title of my project is Different Types of Male Homosexuality Tested Through Different Biomarkers and Psychosocial Sexual Aspects. It's a little bit long, but we'll get through every part of it. So the first thing I would like to talk about is sexual orientation. We consider sexual orientation to be a phenomenon that directs an individual's attraction, sexual attraction, that directs an individual's sexual attraction based on an appearance of other individuals that sends signals of an apparent sexual identity. So usually people feel more attracted to men or women or both. This phenomenon can be broken down into 4 different concepts. The first one would be sexual behavior. The second one would be sexual self-identification. The third one would be sexual attraction. And the fourth one would be sexual arousal. These 4 phenomena, they can be in line with each other, but they aren't always. We have many examples of people who identify as heterosexual and still engage into homosexual behavior, for example. For my project, we are focusing on individuals who identify as non-heterosexual males. So it includes homosexuals, bisexuals, and pansexuals, yes. Another important part of my project are the homosexual subcultures. So we have basically cultural niches of homosexual people, and it makes clear how homosexual people are not an homogeneous group as it was once considered in the scientific literature. So we have many different groups who differ from each other into social rules, values, behaviors, stereotypes, and they have very different patterns of behavior and of their existence. They use very different drugs, for example. They have different patterns of sexual behavior. They have different appearances, and they go to different places. They behave differently. They have different personalities. And this is why I defend that the homosexual population is not as homogeneous as it was once considered. So here I bring you some examples of the most famous subcultures we have in the homosexual community. We have bears, drag queens, twinks, a bunch of fitness subcultures. There must be like 5 or 6. The suburbans, the activists, the alternatives, the leathermen, the daddies, the clubbers, the geeks and gamers, the BDSM practitioners, and the granolas. These are only a few examples. There is a huge list of potential subcultures, and unfortunately, we don't have time to be more detailed in each and every one of them. Another important part of my project are the biomarkers of sexuality. These are basically biological traits that are much more frequent in non-heterosexual people than in heterosexual people. And these biomarkers might be markers of different developmental processes. These processes might be endocrinological, immunological, and genetic processes. And they seem to influence the type of homosexuality that will be expressed in the individual. There are studies already indicating, for example, that people who carry different biomarkers of sexuality express different traits regarding masculinity and femininity, sexual preferences, especially when it regards the penetrative role. When people have to choose if they want to be a top or a bottom, there might be a biological influence there. Choose between many commas. And gender conformity as well seems to have an influence on what type of biomarker these people carry. So the 3 types of biomarker of sexuality that I like to highlight from my project are the fraternal birth order, which basically describes the fact that homosexual men tend to have many more older brothers than heterosexual men. There is a long and complicated immunological theory called the maternal immunity theory behind it. So basically, each time a woman generates a male fetus in her womb, that environment of the womb changes for the next male fetus that will be generated there, and enhances the chance that that fetus, when reaching a mature age, will not be heterosexual. The second one is handedness, which describes the fact that homosexual men tend to be much more left-handed than heterosexual men. And the third one is heritability, which describes the fact that homosexuality tends to cluster in families. So, going on to my objectives and hypotheses of the project, for your better understanding, I have divided the project into 2 phases. Phase 1 would be understanding the correlation between the belonging to different subcultures and the carrying of some psychological, social, and sexual traits. For example, sexual preferences, self-esteem, how people can relate and connect to the LGBT community. What else? Social sexuality, which was mentioned in one of the previous talks. So we have selected some psycho, social, sexual traits to correlate with the belonging to different subcultures in the first phase of the project. The second phase of the project will be correlating these 2 previous variables to the biomarkers. So I intend on separating my participants into 4 clusters according to the biomarker that they carry or to the absence of any biomarker whatsoever, and try to see if the carrying of different biomarkers may be able to predict any other of these other 2 variables. The methods for my project. So we are looking for male people over 18, identifying as non-heterosexual, and when I say male people, I mean both cisgender and transgender male people. For the preliminary analysis of my data, I have used the first 220 participants. So far, we have already more than 720 participants. Our goal is to reach 1,500 of participants. So we're halfway there. The instruments I'm using to collect this data is basically a battery of questionnaires and scales and inventories. One for social demographic information, one for sexual orientation information, one for this gay peer crowd questionnaire is a questionnaire to index whether people belong or not to any of the subcultures that I'm studying. The revised social sexual orientation inventory is to study people's social sexuality. The childhood gender nonconformity scale to assess childhood gender nonconformity. A self-esteem scale and a masculinity/femininity inventory. So these are the psychological, social, and sexual variables that I intend to analyze together with the belonging to different subcultures and the carrying of different biomarkers. As I said, my project is still in a very preliminary phase, so I don't have many results to show you, but I will show you what I have so far. So I made a bunch of Pearson's correlations between all the variables that I'm studying so far. We have not found any significant variation, and therefore we haven't gone much further with the analysis of these tests so far. But since we have already tripled our sample, a new analysis of this data might already have a totally different face than this. Here I'm showing the face of the subcultures in terms of the psychological and social variables that I'm studying. So this one is separating the participants according to the subculture they belong to, and these violent plots show their childhood gender nonconformity. So the scientific literature says that a few of them should express more childhood gender nonconformity than the other ones. So far we haven't found any statistical significance, but that's just a detail. So this is the same graphic, but for social sexuality. The bears, for example, according to the literature, should be one of the subcultures with the highest social sexuality. So far, we haven't found any significant differences, but we still expect to. This is the same graphic, but for self-esteem. The same one for masculinity. And the same one for femininity. So far, we cannot see any statistical differences between these subcultures. But as I said, our project is still at very preliminary phases, and I have very high expectations for the future of the project. So I'd like to thank you, and if you have any questions, I'm all here for it. Tem microfone aí embaixo? Acabou a bateria?
Não. Cadê? Oh. Você tem outro? Ah, tá aqui? Ah.
Tem outro?
Question, comment. 1, 2, 3.
Hi, thank you for your presentation. You mentioned something about homosexual men and left-handed.
Yes.
There isn't something explanation for that. I don't know.
There are hypotheses. So the hypothesis behind the biomarker of handedness is that there may be components that are common between the definition of a sexual orientation and the lateralization of the brain. So there might be components in common between these two processes that might create this correlation. But so far it's just a hypothesis. There's not many data about it.
First of all, thank you for your presentation. I would like to ask if your research if you consider when you're searching about gender and attraction, about the construction between gender and the body, like ciscentrism. I don't know if you are familiar with this.
I'm not sure
concept.
I'm not sure I understand the question.
Like the construction of gender and attraction to the gender by itself, and not the body that is considered like when you construct a gender. I'm sorry. Socially, the gender is seen as intrinsic to the body in a general way. So, like when someone's transgender or non-binary, they tend to the prejudice tend to be about the non-conforming body. So the attraction is built up on the gender, but also on the body. Does your study see these questions as important, as relevant? I don't know.
Are you talking about one of my first slides when I was defining sexual orientation?
Yeah, and the methods.
Now I understand. Well, sexual attraction isn't really the main focus of the project. However, I'm not sure I believe that the gender is something intrinsic to the person. I tend to consider gender a social construct. Sexual orientation, however, seems to have a more solid biologic base. So I'm not trying to biologize gender. I'm just trying to biologize sexual orientation. And as regards to transsexual people and non-binary people, I think our times are changing faster than the scientific literature can keep up, and there's still a lot of data missing on these people. Transsexual males are basically invisible to the scientific literature. There's very few papers on them, for example. And however, I do consider that when people transition on their gender, their behavior might or might not align with their gender identity or with their sexual, or with their biological sex. Depending on the case, depending on whether the person does or not hormone treatment, all of these factors can change a person's behavior. So it's a little more difficult to get there than we wish it were.
Hi. Here. About the biomarkers, does it apply to transgender men, trans men?
No. Only cisgender men and transgender women. Yes, because a part of it seems to come from an interaction between the Y chromosome and the mother's body.
So you said you're considering trans men also in your research, so you have to, it's going to interfere.
Yes, I do believe that the participation of trans men in my research would be very useful, especially regarding the belonging to the subcultures, because we don't have information about how these people interact with the subcultures yet.
Okay, thank you.
So I think this is where the trans men would be particularly important for my research.
My graduate student and I are looking at I'm thinking of some additional biomarkers that you might consider. My graduate student and I are looking at anthropometric traits like grip strength that are highly sexually dimorphic. and looking at the relationship between those traits and sexual behavior. And our interest is primarily in heterosexual, but just out of curiosity, we looked at the relationship between these traits and homosexual behavior. We have a nationally representative sample with, I think, about 5,000 people in it, and we have about 50 to 100 homosexuals in the sample. And what we saw was that male homosexuals were shifted kind of in the female direction anthropometrically, and female homosexuals were shifted in the male direction anthropometrically. And our sample sizes are probably too small for us to be confident enough this is real, but it's conceivable that might be another interesting biomarker, are these highly sexually dimorphic traits might somehow also be related to homosexual.
Absolutely. There are a few other biomarkers in the literature. For example, the second to fourth finger ratio, the ratio between the sizes of the fingers. However, since the instruments we're using for the project are basically questionnaires, we decided to focus on the biomarkers that would be possible to assess through these instruments.
Right.
So, for example, the heritability of homosexuality can be accessed through asking people if they know any other homosexuals in their families. I know this is not a perfect method, but it's how we're doing so far. And so these are the biomarkers we're focusing on, basically because they are the most possible ones to be assessed through questionnaires and scales, which are our main instruments. This project was designed to be carried on during the pandemics of COVID. So it was supposed to happen online.
Right. Yeah, the data sets we're using are public, and they've been conducted, they're nationally representative, so, and they do have a lot of these anthropometrics and sexual behavior over many years, so there might be enough data in there for a new project at some later time.
Absolutely.
Yeah.
Thank you very much. Thank you, Bruno. Thank you, everybody, for this very interesting day and presentations and discussions. And that's it for today. Don't forget to come tomorrow, and don't forget to come tonight for the banquet, so that we can continue the discussions in a more unofficial manner. So thank you very much again. It was very nice.